Good morning, good afternoon, good evening, everybody, depending on where you're joining us from. My name is Sue Lynch, and I am delighted to welcome you here today to our annual symposium at the Benioff Center for Microbiome Medicine. One of the impacts of the global pandemic is our inability to meet in person for this symposium. This is our second symposium. But by virtue of going to a virtual platform, the silver lining is that we have more than 400 registrants from across the globe. So beyond the United States and Canada, we would really like to extend a warm welcome to attendees in Europe, South Africa, and Asia. We're absolutely delighted to have you here with us today. And I'm really excited for the lineup that we have. It promises to be a very exciting symposium. And before we get started, I'd just like to say a few words about the field and about our new center here at UCSF. I know that all of you are well aware that in a few short years since the inception of the field of human microbiome research, we've really advanced in leaps and bounds. From the earliest observations that our bodies are home to a thriving, diverse ecosystem of microbes, to the observations that these microbes strongly influence our health and well-being. The field has really developed rather rapidly, and the excitement and the expectation is rather large, I will say. And a number of the findings that have been made in the field have been made here on campus at UCSF. Everything from the observation that diet plays a large role in shaping the types and activities of microbes in the human system, to the observation that drug bioavailability is influenced by microbial species and specific strains that metabolize these ingested pharmaceuticals. Other findings from faculty across campus include the observation that very early life microbial encounters in the skin and in the gut shape immune function. And these studies have led to the observation that very early perturbations in the gut microbiome, for example, relate to the development of childhood diseases like asthma and allergy. But these studies have pushed past correlations, and they've allowed us insights into the molecular basis of immune dysfunction in very early life that drive disease development later in childhood. And it's these and many other studies that have gone on across campus, as well as the unfailing support from our leadership and our development office, that allowed us to secure an incredibly generous gift from Mark and Lynn Benioff to launch the UCSF Benioff Center for Microbiome Medicine. The center is home to a diverse community of investigators across campus that are invested in leveraging this field to predict, prevent, and treat disease. And though we are really a fledgling program, we launched in the summer of 2019, we already have over 100 members in our community, including faculty, staff, and trainees across all 4 schools on campus: School of Dentistry, Nursing, Medicine, and Pharmacy. These engaged individuals participate in our educational programming, our monthly seminar series, which is open to members of the public and investigators beyond UCSF. We also have a summer internship program for disadvantaged youth in the Bay Area. And we really believe in building community and building bridges, not just in UCSF, but across institutions nationwide and globally. Our research program, our first research funding announcement, was launched in the spring of last year and led to support for 14 investigators across campus. And as you can see from the icons here, the transcendency of the field. Our investigators are spread across a number of departments and programs across campus, really speaking to the diversity of interest and research that's going on campus in the microbiome field. We also have supported the Tri-Institutional Microbiome Partnership. This has been an initiative to support interdisciplinary research across institutions, and involves UCSF, UC Davis, and the National Microbiome Data Collaborative. And in an effort to build new and expand existing programs on campus, we've partnered with leaders of the California Preterm Birth Initiative and the International Multiple Sclerosis Microbiome Study, as I said, to expand and to develop new microbiome research efforts in these arenas. All of our studies and all of our investigations require investment and in technologies. And we recognized early on that this can actually be a barrier to many getting into the field of human microbiome research. And so we've invested in what we call collab incubators on campus. These are not service provision facilities, rather they're collaborative labs led by domain experts, which allow for collaborative studies and interdisciplinary studies between members of these collabs and investigators across campus. And ultimately, we'd like to make these facilities available to others outside of UCSF. We focused on collab incubators in the arenas of microbial genomics, metabolomics, and small molecule discovery, microbial culture, and we're expanding our existing notobiotic mouse facility. We feel these are the tools that are required for the field, and that by standardizing our efforts in these arenas and offering embedded training and innovating in these fields, we'll really accelerate our efforts in research in the field. And we're really grateful to our sister program, ImmunoX, on campus for their co-sponsorship of many of these collab incubators. And finally, obviously, none of this would happen without our community and the strong leadership of an incredible group of faculty that I'm just privileged to work with, our executive committee and center management team. I'd especially like to thank Sergio, Katie, Tiffany, Peter, Kristen, and Sarah for all of their efforts and allowing us to develop our center over the last 18 months. We're just thrilled with our progress and our community building efforts on campus. And I would especially like to thank Peter, Kristen, and Sarah, whose tireless efforts have brought you this symposium today. They've really done an incredible job. And that's enough about us. Let's get on to the agenda for the day. I am just absolutely thrilled and very grateful to each and every one of our speakers for agreeing to join us today. I know that there's a lot of demands on everybody's time, and I'm just thrilled to have the key leaders in the field present to you today. So how this will work is that each of our executive committees will moderate these sessions. You can see the agenda through the link provided here at the bottom of your screen. And if you have any questions for our speakers, we ask that you provide those questions through the Q&A facility, through the chat facility on your dashboard. And so with that, I'd like to hand over to Dr. Sergio Baronzini, who will introduce our keynote speaker. Sergio.
Thank you, Sue. Welcome, everybody. It is my honor and pleasure to introduce today our keynote speaker, Dr. Jeff Gordon. He was an inspiration to many scientists, including myself, to get into this field, so it's a real pleasure. Dr. Gordon is joining us from Washington University at St. Louis, where he's the Robert Glaser Distinguished University Professor and the Director of the Edison Family Center for Genome Sciences and Systems Biology. He's been, I think, appropriately dubbed the father of microbiome research. He and his team were the first to demonstrate the importance of the gut microbiome in regulating animal physiology. Dr. Gordon's work has shown that the microbiota performs specific and essential functions without which the human cannot work as it should. He has also shown that the long-term effects of childhood malnutrition are determined not only by diet, but also by the presence or absence of a healthy microbiome. His work has enabled new research directions in the study of multiple conditions and the search for novel treatments. He has received countless national and international awards, holds over 20 US patents, and he's a member of the National Academy of Sciences, American Academy of Arts and Sciences, the National Academy of Medicine, and the American Philosophical Society. Dr. Gordon's lab is currently focused on developing gut microbial community-targeted therapeutics to treat undernutrition in infants and children living in low-income countries and obesity in westernized countries. The title of his talk is Development of Microbiota-Directed Complementary Foods for Treatment of Childhood Undernutrition. Let me now move to our next speaker for today. Next speaker is Ruth Ley. Dr. Ley is a professor and director of the Department of Microbiome Science at the Max Planck Institute for Developmental Biology, and a speaker for the Cluster of Excellence Controlling Microbiomes to Fight Infection with the University of Tübingen in Germany. Dr. Ley is also on the Scientific Council of the Institut Pasteur. Dr. Ley has received numerous awards and is a member of the EMBO, and the European Academy of Microbiology, American Academy of Microbiology, the Leopoldina German National Academy of Sciences, and most recently was a recipient of the 2020 Otto Bayer Award. Dr. Ley's lab is currently investigating the evolutionary history of gut microbes and how they have adapted to life inside humans, and now how they affect human biology and health. So the title of her talk is Linking Vertical Transmission of Gut Microbes with Their Patterns of Co-phylogeny with Human Hosts. Please, Ruth, floor is yours.
Okay, can you hear me?
Yes, we can.
You can see, all right. Well, thank you very, very much for the invitation to speak. I'm sad. I'm very sad that I can't be in San Francisco with you guys. I'm very grateful for Pete to have invited me to be there along with Jeff. As you know, Pete and Jeff are the reason I've been really able to have an independent research career, because mine was launched working with both of them back in St. Louis. And so I was really looking forward to meeting up with everybody and meeting new people and talking about these topics, but that can't be. So instead, I'm speaking to you from Germany today, and but I'm happy to share with you some of the work we've been doing on co-diversification in human hosts. And I'm going to start off with an anecdote, if you will, of vertical transmission, and this is work that was started in St. Louis, and I'll show you what I mean. So I think Jeff has laid the stage beautifully for why the infant gut microbiota can be so important for the life of the person and the development of the person. And there's been a lot of work from the community over the last 10 years or so, and increasingly now that we have these really powerful techniques such as metagenomics and the ability to track strains, there's more and more work coming out showing where these strains might come from and which ones might come from the mother, for instance. So on the left side of this slide, there's a graphical abstract from a paper from the Segato lab in which they looked at transference of strains from the mother to the infants, and after about 4 months, you could pick up strains that clearly came from the mother's stool, for instance. And then there's other works out of Per Bork's lab recently where they looked within families at similarities between strains, and there they've looked at kids of different ages in relation to their mothers, for instance, or with each other. And where you could start seeing some perhaps stability within individuals, that the similarities with the family members might drop off with time. So there's still some questions open about how long strains persist in individuals, and particularly strains that might be passed down from parents or within families. And so I think as a field, we're getting a better idea, at least in some populations, of what microbiomes look like in newborns, in early childhood, in adulthood. And these are coming from separate cohorts. And what we don't have, of course, is longitudinal data that might have come from single individuals over lifetimes, and that's simply because we haven't been around long enough to have put these sample sets together. So how long strains actually persist and how much they track in families is still to some degree open questions, but we're starting to get some really intriguing hints. So I'm going to start by telling you about a follow-up of this paper. So this paper was the first paper out of my lab. And so we weren't very humble with the title. Like Jeff says, we should be humble. We called this Succession of Microbial Consortia in the Developing Gut Microbiome. In fact, it was one, and it was a case study. But what was, I think, intriguing to people about this was that this was a single individual that was sampled quite densely over the first year of life, and then a bit longer after that into the second year. And what you're looking at here is the succession of the phyla over time. And then we also had information on baby M about, for instance, when he started on table foods, and it was right after that you saw a big increase in the Bacteroidetes, for instance. And so this was telling us that some of these big swings that you can see in an infant gut microbiome developing over time can occasionally be explained by things that you might know about the baby. And so that's where we were at that point. And what I want to do now is give you a 10-year update on baby M. And this was actually kind of stimulated by meeting up with Jeff and Pete again, because this study really did have its roots back in St. Louis. So what did we do? We resampled this individual 10 years later. And as far as I know, I think this might be the only baby that has been resampled 10 years later. So even though it's an anecdote and a case study, it's kind of, it has some intriguing findings, and maybe at some point we'll have more data on this kind of thing. So this was IRB approved, but working with a teenager can be kind of challenging because the subject now talks and says things like, this is worth more to you than it is to me. So there's some negotiation that has to go on when you're dealing with this age group. But the parents were perfectly eager to be sampled. And so what we did, along with Liam, who's a graduate student in the lab, is go ahead and resequence baby M and teenager M samples and assemble these metagenomes, and using a Synteny tool that was developed by Hage, a postdoc in the lab, we're able to start tracking these strains through baby M's time into teenager M and comparing with the parents. So what happened in the past 10 years? Of course, this is all anecdotal again, but diet, of course, is something that changes and it becomes more and more uncontrollable in the subject. This subject was lucky enough to really only have mild illnesses, such as norovirus, which he coincidentally had on a flight from Munich to San Francisco over an 11-hour period. And I think in that case, you might see strains shared with other passengers in a unilateral direction. But this individual also changed continents, and this brought about some behavioral issues, maybe like telling people from the host country who won the war. And maybe, you know, there's differences in social contact when you have this kind of attitude, and maybe that has effects on strain sharing. The point is, we'll never know. But here he is, gotten quite tall, striking similarity to the phenotype of the parent. Is that due to strain sharing? Again, N of 1, we'll never know. So getting to the data, we've shotgun sequenced baby M and teenager M. And it's nice to see that we basically have the same pattern again. We have table foods coming in and driving the Bacteroidetes. What we weren't able to see last time because of the primer set that we used, we have more actinobacteria in the middle here of the first year. And as baby M turns into a teenager, you can see a pretty typical adult pattern of firmicutes and Bacteroidetes settling in. The diversity has been going up so these are basically species level diversity from the metagenomes. You can see an increase over time. Teenager M is up here. And what we're showing with the red and the blue is which samples we pulled for strain comparison for the purpose of this talk. So the Synteny-based tool that Haggai developed takes reference genomes of various taxa, compares them to per sample metagenomic assemblies, makes collections of 5 kb regions for each sample, and then using k-mer identities identifies these Synteny blocks that you can then use to assign a Synteny score. So basically at the end what you have is that for each species that you detect in pairwise samples comparisons, you can get an idea based on the Synteny of those genome pieces, just how similar those strains were. And Haggai has benchmarked this against data that came out last year that involved longitudinal gut metagenomes for many individuals and different bacterial species. And what you can do then is set a strain sharing cutoff based on the Synteny, where if you have a Synteny score above this certain line, then we're going to call that the same strain. So this is within individual, this is between individuals, and these would be considered the same strain. So just to give you some faith that this works, we have used this in a paper that we published last year to look at bifidobacterium strains in twins that had different genotypes. We were looking for strain differences between people with different genotypes at the LCT locus. We didn't see any there, but if you look within individuals or within twins compared to unrelated, you'll get different Synteny scores that are telling you that people have the same strains of themselves sampled over time. Twins will share strains and unrelated people less likely to. So we've also compared it to what you get with standard tools such as StrainFlan, and it gives you basically the same kind of output. So now we take subject M and compare subject M to the mother who was sampled at the same year. The mother sampled 10 years later and the father sampled 10 years later. And these are baby M's samples arranged along a time axis here from young to older. And whenever there's a black dot, it means it was the same strain. And if it was a yellow dot, it's a different strain. And so I'm showing you this first just so that you can get an idea of the list of things. There's interesting things in here. There's Methanobrevibacter smithii showing up. There's a number of things, and I'm going to walk you through some of them. Okay, so we don't spend too much time on this slide. So Bifidobacterium longum. We're going to look now at within baby M over time. This is the age gradient against itself, so that you'll see the diagonal is Synteny of identical strains. And if you look here, if you look basically, if you look along the bottom, you're going to see the younger strains getting compared to the older and older strains. And you see this really nice gradient from blue down to orange, which is showing you that there's this slow change in the strain over time. So down here is comparing teenager M to baby M, and it looks like strain replacement at that point. So now instead of showing you this, I'm going to convert this to what it looks like if we do strain calling and say yes, no, same strain, different strain, and it looks like this. So you can see that the early baby days had the same strain, the teenager has the same strains, and there's some time in the middle where there was the same strains, and for all we know, these are now different ones. So we can't compare this to the parents because this one we didn't pick up in the parents. But now let's look at Bacteroides uniformis. So Bacteroides uniformis was the same strain for the first 2.5 years, and then it's a different one 10 years later. With some exceptions, because of these are very high Synteny scores. This is called as the same strain that's been there from the beginning. So in Bacteroides uniformis, it looks like we can find what looks like the same strain back then all the way through, and then it's still there in some of the samples today. Now, if we compare to the mother and the father, you can see that the teenager and the mother are sharing a strain that the mother had 10 years ago, that the mother has now, And then baby M also had sharing with the father and sharing with the mother. So there's sharing all the way across here. And this is really telling us that these strains are shared by the whole family and persistent over this 10-year period. Barnesiella intestini hominis also came up as an interesting one, where we see sharing all through the baby years, and then we see that the teenager has some stability. And if we convert that to yes, no, same strain, different strain, we can see that this one is also consistent all the way through. So we still find the same strain as a baby, as a teenager. And here, these strains were really shared by the family over the course of the 10 years. So you could see mother and father 10 years later sharing strains with the baby and the teen. Let's now look within the Bacteroidetes. We've got data on Parabacteroides distosonis. So in this one, you can see that the baby is sharing strains with that were detected in the mother 10 years apart, but nothing with the father. Nothing. This one's just shared with the mother. This one, B theta, it's shared with the father during the baby years, and there's absolutely no sharing anymore with anybody. This one has been, Bacteroides cellulose has been shared with the mother the whole time and even now. So this looks like the mother has been sharing the strain with baby M and teenager M probably the whole time. At least that's what we're seeing with a gap. And then here's something a little different. These are again in the Bacteroidetes families. These are Elistopes. We didn't see these in the baby at all, but the teenager has them. And for Elistopes putridinus, the whole family has the same strain. And for Elistopes shahi, clearly the teen has a strain that the rest of the family doesn't have. So the 10-year update on this study is showing us that in this case study, we see strains that are persistent in the first 2 years. We see strains that are persistent over 10 years. We see strains that are shared with the mother. We see strains that are shared with the father. We see strains that are shared by all three. And we see new strains appearing after 10 years. So some of everything, basically. So from this N of 1, we can say that at least in this person, what this person was seeded with at the very beginning has stayed with this individual for at least 10 years through changes in diet, through changes in environment, through some illness. They're still there, and so whether or not this has implications for the health impact of these strains is something that I guess as a field we're still figuring out in general. But this does lend more weight to this idea that this transmission of strains that's occurring over generations. And with that, we can extrapolate out and maybe think over very long periods of time and about how humans have dispersed out of Africa to colonize the world in waves and in ways that we know about through genetic comparisons of humans and with the fossil record and the anthropological record. It's clear that and we have a fairly good understanding now of when people came out of Africa in waves and where they went at what times. And what's been fascinating from the standpoint of microbiology is that we know that we've carried Helicobacter with us during this process. So this was shown beautifully by the H. pylori researchers who looked at strain comparisons based on cultures of H. pylori and looked at the different strains around the world and saw that they actually were consistent with the out of Africa pattern of colonization. So we've wondered really, besides H. pylori, what other gut microbes have co-diversified with humans? Because it seems unlikely that H. pylori is the only one. And can we get at that with metagenomes now? So what we'd want to do then is test for co-phylogeny by making trees of host genotype and building individual trees of bacterial strains, and then asking how well do they match. And if we get a really nice match like this, this would show that there's been co-diversification. And if we get more of a mixed signal, this would mean that there's been frequent exchange between hosts over time and between populations, and that the signal is really erased. We know that this is happening at the larger vertebrates level. So for instance, Nick Youngblood in my lab, who works with me, has looked at a large collection of wild vertebrates and asked these questions using 16S ribosomal RNA genes, and sees that within the mammals and particularly within the herbivores, we have signal from the data that supports this kind of pattern rather than this kind of pattern. And then if you start looking at the hominids, this was work by Andrew Mueller and Howard Auckman, and they showed very nicely using a single gene, this time diarase B, that the bifidobacteria and some of the Bacteroides had patterns that matched the phylogenetic patterns of their hosts. And so here there could have been a strain transfer from bonobos into gorillas, but generally this pattern is consistent with what you see in the host, and this is pretty strong indication that the bifidos have actually been following their hosts too over time, and are so adapted to them that you can see these strains in the data now, these patterns in the data. So besides these, and besides H. pylori, what other bacteria might track with human genotype across populations? And so to answer this question, what we need is metagenomes in which we can track strains, and there's hints already. So for instance, Nicola Sagada's lab that developed StrainFlan for tracking strains in populations has found that within species, different populations have distinct strains. For instance, from their work, the bifidobacteria, again, there's different strains, and you'll find them at different proportions in different parts of the world where you look. But we need, in order to match with the human trees, we need to build human trees out of human genomes, and we need matching gut metagenomes, and these data sets are hard to come by. So we went out and generated some of this data as part of a study that we were doing on lactose tolerance. In Germany, in Gabon, and in Vietnam, we sampled a number of mothers and their children, and we obtained from the mothers genetic information as well. So Taichi Suzuki has been working on this, and he's working with 630 adult female subjects and their gut metagenomes and their genotypes that he's obtained, so from our collection, and adding in the UK twins publicly available data for which we have similar data set. And then, so what he's been able to do is from the human genomes, using SNP phyllo and using about 13,000 SNPs, he can build a host tree. And from the metagenomes, he's applied StrainFlan in this case and 200 marker genes. And for 53 microbial species that are observed in at least 100 individuals, he can build those trees. And so now what he needs to do is compare for all 53 the host tree to the microbial tree. And then within these populations, within the Asian, African, and European populations, he can also ask the same question within these populations as well. And so to compare these trees, he's used PhyTools, he's used Parafit, and he's used PACO, and these are different ways of asking how well do these trees match. So this is what the human tree looks like. These are now the human hosts. Based on 13,000 SNPs, putting them all together, you can see these are this African population, this is the Asian population, and this here is the European population. So these are the continents. And then on the outer ring, the added information of whether they were UK in green or Germany in orange. So we have three nice big clusters of people that separate out from each other a little bit based on their SNPs. And now these are some of the top taxa and some of their patterns. And so you're going to be comparing back to the human tree here. And so now these are trees that are built using strain data. And this is Provotella copri. You can see a nice clustering by the African origin here, and some Asian here, and the European. And in fact, when you see different clumps like this, we're probably looking at different strains. And if you look within each strain, you can see that each time the most basal strain is actually the African strain. So the red, even when you have a new cluster, the red is typically the African strain. Over in Ruminococcus, it's the same. There's the African strain, Asian. Here we have a lot more Europeans represented. So Provotella copri, we didn't find so much in the Europeans. And then here you see a lot of the Europeans represented with the English and the German people all mixed up. No separation with them there. Here's Eubacterium rictali and Eubacterium elegans. Same kind of pattern, starting with Africa basal, Asia, Africa basal. Here's some Asia, some more Africa, and then we have Europe here. Again, the mixed Germans and English people. Same kind of pattern with Eubacterium. So we have 53 of these. The point now is to ask how similar is it to the human tree, and is that by chance or not? So this is what the data compiled look like. Here are the 15 top taxa and their names. And then here's the total number of people represented in each one of these that have data for each one of these taxa. And so the numbers vary because of the depth of sampling and because of the abundance of the particular taxa. And then what Taichi's done is run these 3 tests twice. In this column, he's run it using a tree where the branches are collapsed if there's no bootstrap support. And in this one, he's using the best maximum likelihood tree. So, and this is the most conservative column here, and this is how the data is sorted by the most conservative one first. So these are the top 2 that I showed you first, and they're highly significant across multiple tests. Here are the Eubacterium species as well. And so you can see the support for several of these across many of these tests. Of course, there is the possibility that when we see patterns like this, it could be by chance, and it could be because you know, you see distinct sets of strains in distinct populations because of other things, like they have different diets and things like this. And so another way to make sure it isn't just driven by chance and other factors that affect strains, Taichi also ran these analyses within these 3 continents. And so here is the support for the conservative test within Asians, within Africans, even within these populations. We're seeing that on average, there's a non-random chance that you're going to get the strain tree matching the human tree. You'll notice also bifidobacteria showing up in the Europeans only. And bifidobacteria, that tends to be more abundant in the Europeans overall. In order to beef this up, though, we also added in over 1,000 or over 1,300 public metagenomes. And so in this case, what Taichi did was take the public metagenomes and then assign them a human genotype based on what population they were from. So he sort of picked a representative one from around the world based on work by geneticists. And so here are some of the data here as well. Provotella copri, we have strains popping up, and each time the African one is basal, except here. So here we have a section that's clearly Asian, and then there's a big Africa cluster in the middle, and that's from Madagascar. And Madagascar is an interesting human population that's admixed between Africa and Asia. And it looks like from this data that they brought their Provotellas from Asia. So the Madagascar people, although we're labeling them African, actually have an Asian strain, and that might represent their Asian ancestry. Again and again, we're seeing the African strain basal. And then in a few places, we've been able to pull the same species from primate metagenomes, and they're even more basal to the whole set. So that's shown in the pink. So for all of these now, we can also ask, is the basal strain for each one of these clades and each one of these trees Africa or primate? And for 9 out of the 15 top taxa, that what we're seeing is actually consistent with a primate or African origin. So what we have now is a list of gut microbial taxa that look like they've been co-diversifying between and within populations. They're supported by strains from additional human populations and primate species. And we think that this does support very long-term vertical transmission. And so the question then becomes, what do they have to do with our health and our evolution? And so just very quickly, I'll finish up by saying that when we think about these patterns of dispersal around the world, oh, sorry, I lost my train of thought. Let me just finish up by saying that this list of microbial taxa that we got from Taichi's work of co-diversification, we can quickly match it up with some of these that are showing familial sharing and persistence from one place to the next. And this is from an incomplete case study, right? So I think if there's a lot more work to do matching what we see from co-phylogeny with the strains that we're seeing vertically transmitted, and also with the ones that we see responding to host genotype. So these strains that persist in individuals and are transmitted within families are probably transmitted across generations. So they've been with hosts as they disperse, possibly through quite a long period of time. And as people adapted to different places in the world, they probably also are responding to differences in host genotype. And they might even help shape host genotype. So we have some examples of where bifidobacteria, for example, relate to host genotype. And we have examples of where, say, Ruminococcus responds to host genotype. And we know that human genotypes have changed as people spread around the world and adapted to local conditions. And we also know that all of these differences in genotype can be also assigned a change in a microbiome as well. And the challenge is I think for all of us to understand how this fits together, how strains have cross generations adapted with host genotype, and then maybe also helped us adapt to the environmental conditions that we've experienced as we dispersed around the world and as our genotypes changed with those exposures. So with that, I'd like to finish by acknowledging the people who did the work at Teenager M, the study participants, of course, from the other studies as well, and our partners here in Tübingen who've made this possible, particularly Peter Kremsner from the Institute for Tropical Biology and his partners, these who are all medical doctors in their own centers in Vietnam and also in Gabon, who've helped us tremendously with our work. And with that, I'll stop and thank you very much.
Thank you so much, Ruth. That was terrific. We do have time for a couple questions. We have limited time. And I will, so Peter, you wanted to ask if in the first part of the talk, whether the examples that you provided are examples of strain replacement or evolution over time within the host. I guess this is in the first part of your talk.
That's something that we'll be looking into much more closely. So, it's a very good question if we can actually tell how these strains might be changing with time. I can't tell you right now because we just got the data, but that's something we're excited to look at. Great.
There's another question from an anonymous attendee who's interested in, does the idea of co-phylogeny imply that a healthy gut microbiome needs to vary based on the individual genomes? So therapies directed towards the microbiome actually need to be tailored to the individual's genome, or
There could be cases in which that might be the case. I mean, I think we're pretty far from knowing what those might be, but it may be down the line, yeah, depending what you're treating and why. And I've wondered also just how can you dislodge strains that have been persisting in an individual for years? That must be quite a challenge.
Indeed. Okay, so we're right at the top of the hour, and there's a few more questions, but in the interest of time, I'll hand it over to Peter. Thank you so much, Ruth.
Thank you very much.
I'll hand it over to Peter, who will introduce our next speaker. Thank you so much.
Okay, great. Thanks so much, Sergio. It's been a really inspiring and exciting morning already. And I wanted to say to the first two speakers, Jeff and Ruth, if you want to, I think you should be able to like see the remaining questions on the Q&A, so you can type your responses if you have time. But yeah, it's my pleasure to introduce David Mills. He's coming to us from University of California at Davis. And David is really an expert in the bacteria that are found in the early life gut microbiota. He has over 20 years of experience working on gram-positive microbes with an emphasis on lactic acid bacteria and bifidobacteria, used often in food fermentations or as probiotics. And he's really led the area in terms of understanding what these microbes are consuming in the early life gut and what implications that has for our health. And so with that, I don't want to take up too much time, listing all his accolades, but he has, many important titles and awards. And I'll turn you over to him to tell us about the exciting science that he has to share. Welcome, David.
Thank you very much. First, I'll ask if you can hear me.
Yep.
Okay. It's an honor to be here. Susan and Peter, I really appreciate the invitation. It's also an honor to follow both Jeff and Ruth. It is really great to see the work that's ongoing. I'm going to be talking a little bit about how human milk is providing us some lessons that those in the probiotic and bacterial therapy realm might want to learn, and milk might teach us something, and I'm going to follow this theme. First, I'm going to give you my disclosures. Luckily, because of the first two talks, I don't have to spend any time on this particular slide. I think we all understand what a gut microbiota is and all the many wonderful things that it does. But occasionally, it can cause a problem, and we might want to repair it. And I think Jeff gave a wonderful talk on how you might use foods to try to redirect the gut microbiota in a healthy direction. But the question is, do these kinds of, especially if you're trying to transfer microbes back into the gut, do they really work? And this is really not a new concept, of course. Metchnikoff, a long time ago, proposed restoring function in the gut. In this case, he used fermented milks, and hopefully he knew it would deliver microbes at the same time to resolve what he termed dysbioses. And this is a long time ago. And just as now, then they had big critics. And if you read Paul Groof's Microbe Hunters, he criticized the many companies that were spawned from Metchnikoff's work. And I love this line, and their directors grew rich off selling these silly bacilli. You could make an argument that we have a similar situation today in that we have lots of products aimed at the gut, and they're all hoping to make a change, a beneficial change in the gut. There's traditional probiotics on the left that have been around for a while, and of course, we have with the new advance in microbiome science, we have lots of new live bacterial therapeutics that companies are increasingly beginning to apply. But there are some strategies and some concerns that have been brought out. And this one is a nice perspective by Siegel and Elenov that came out in Nature Medicine recently, talking about some of the problems, at least with current probiotics. And I would argue this could stretch into some of the live bacterial therapies as well. And one of the things that they noticed in a reviewing a variety of probiotic trials is that there's an unresolved issue in that, do the organisms that you're feeding actually colonize? Do they colonize in reasonable numbers? Do they colonize the gut mucosa? Do they stick around for any period of time? Most folks who've studied probiotics know that most probiotics, certainly off-the-shelf probiotics you can buy now, go through you rather quickly and stay at a reasonably low level. In other words, they can't override colonization resistance. And therefore, the question that the authors here had was, is that part of the fact that we don't see current probiotics as effective as perhaps they could be? In other words, are they not colonizing? We just need to get them in higher numbers and get them to persist, maybe in a tunable way, so that we can have an effect. So if you look around for natural models of this, there's really one natural model of delivering microbes, feeding those microbes, and then having those microbes go away. And that, of course, is milk and nursing. So this is the diet-based enrichment of a microbiome. And of course, when you wean, as Jeff showed, you end up with a different microbiome. So this is a classic profile of infants in early stage of life. And the microbiome development. This is a nice review from a couple of years ago by Arietta. And it shows that you're born, as I like to say, reasonably sterile. And then you acquire from mother and the environment a microbiome that often in the early stages of breastfeeding is dominated by bifidobacteria, but then starts to diversify as you get older. I would argue this profile here represents more of the industrialized world. There's parts of the world where you'll see during breastfeeding really dramatic increases in bifidobacteria, much larger than what you might see in the industrialized world. But of course, once you wean, once you move on to other food, that big bifidobacterial population goes away. So we really have a diet-based enrichment of specific taxa. Taxa that are well known to be associated with the health of the infant. So we wanted to figure out what is driving that. You really need to look at the components of milk. You've got to get inside the bioactive molecules, as Jeff was talking about before, of the food. In this case, it's going to be breast milk. So what are the components of milk components that are influencing the infant gut microbiota? And more importantly, do these components act in a biased way? Do they enrich some and restrict others? So I'm gonna present milk somewhat simply here in three buckets. And those three buckets might be antimicrobial activities, host modifications, and prebiotic enrichments. We know that milk does a variety of host modifications. Direct milk bioactive molecules go in and modify the host. I'm not gonna have time to go into that on how it might shape the microbiota. But we certainly know a lot about antimicrobial activities as well. And this is a partial list of many of the antimicrobial activities that are present in milk. Some of them are quite famous, of course, lysozyme and lactoferrin, et cetera, immunoglobulins. But what you might not know is a variety of these are actually quite biased in their direction. Lysozyme is well known not to work as well on infant-born bifidobacteria, and there's been a series of publications that demonstrate that. In our lab, working with Carlito Labria and Bruce German, Ishita Shah has demonstrated that there are antimicrobial peptides naturally present in milk that seem to kill everything else except bifidobacteria and lactobacilli. And so there's bias in some of the antimicrobial peptides that might be produced in milk. And I want to tell a short story about immunoglobulins. So this is some work that was done by Vanessa Castagna in my lab and also in Bo Launerdal's lab here at UC Davis. And she was focused on secretory IgA. So secretory IgA, of course, is delivered in milk. It's one of the predominant IGs delivered in milk. And its main role, of course, as we assume from an IgA, is to bind a pathogen and deliver it to its doom. But we can't forget that secretory IgA, like many milk proteins, are glycosylated, in this case, heavily glycosylated. So this is decorated by a whole bunch of both N and O glycosylations. And Blaise Corsese previously had shown that some of the interactions of at least gram-positives with immunoglobulin, secretory immunoglobulin A, is through that glycan part, not the binding pocket of the Ig. So we were curious, because work in my lab had previously shown if you grow B. infantis, for instance, or Bifidobacterium infantis on HMOs, it binds to the glycans on CACO2 cells better. And so we thought, well, it must be binding also to glycoproteins in milk if they're co-resident. And so this is what Vanessa has worked on in the past couple of years. And she showed that by some flow cytometry that you can just simply bind B. infantis, it's B. longum subspecies infantis, to Sig-A. And of course, as you increase the amount of Sig-A, you see a wonderful binding between the two. And you could imagine this happening in freshly expressed milk. Is this part of all the milk microbiome that interacts in the milk that gets delivered into the child? There's going to be an interaction with that milk microbiome with the glycoproteins and the glycolipids and the HMOs in milk. So maybe this is part of an interaction that occurs early in delivery into the child. If so, does it protect in any way? So you might say, well, maybe this binding protects from digestion. And so Vanessa looked at in vitro gut models of digestion, and sure enough, when you pre-bind B. infantis to Sig-A, it protects it from a mock gut digestion. Ironically, it also assists in binding to CACO2 cells in an in vitro fashion. So now we have potentially protecting the cell, perhaps delivering it to the intestine in a more robust fashion. And you say, well, wait a minute, does this have anything to do in vivo? Because these are all in vitro tests. And so Vanessa did a really clever mouse experiment where she simply gavaged a mouse with either B. longum infantis or B. longum infantis that had been pre-bound to milk Sig-A, and only provided it for 3 days, and then just asked, how long does it stick around? And she looked at B. infantis, in this case, persistence, post stopping giving the organism. Most probiotic studies continue to give it throughout the treatment. Here we're trying to assess persistence. Now, when the B. infantis was given by itself, you can see over here, you can see it in day 4, right? A couple of days after you stopped providing the probiotic, but it goes away. You can't see it anymore by qPCR or by CFU analysis pretty quickly, like most probiotics that are given to people. They go away pretty quickly. Conversely, if you pre-bind it to Sig-A, we could continue to see it out, and we happen to end the study on day 9. We both wonder how long this might have actually lasted out. So perhaps we're getting a increased persistence in delivery of an exogenous microbe by pre-binding it with a glycoprotein like Sig-A. All right, so we'll go back to my 3 buckets again. This suggests you can imagine a situation with the IgA data that I just showed you, where it is binding select organisms like the saccharolytic organisms and not binding other organisms. So those organisms that might consume mucin-like structures would be preferentially selected. But what about prebiotic enrichment? So of course, prebiotic enrichment is a substrate that is selectively utilized by the host. Microorganisms conferring a health benefit. In milk, there are a lot of glycans. There's free human milk oligosaccharides, and this is depicted here by this structure. This is not a structure that actually exists. This just represents all the types of linkages and all the monomers that are present in human milk oligosaccharides. And of course, those structures can be connected to lipids or proteins. And so there's a lot of glycans that are delivered. If you're a microbe and you want to eat this, you got to have all of these enzymes available, docked either inside or outside of your cell if you want to try to cleave this and consume it. And my lab and many other labs now from around the world, and I've just listed some of the flags of the countries here, have been studying consumption of human milk oligosaccharides by different bifidobacteria. So here's a pool of human milk oligosaccharides. In this case, it's a bubble plot where all the ones in red have a fucose on it, the ones in yellow have a sialic acid on it, and the ones in white have neither. And this represents a secretor mother pool of milk oligosaccharides. If you look at the infant-born bifidobacteria that are commonly examined and have been examined by researchers in these various countries, in general, B. longum subspecies infantis is the only one that actually consumes the whole pool. Everybody else eats a portion of that pool, and gets at a point that Jeff was commenting on before about how the bifidobacteria, the relationships between different bifidobacterial species in the assembly of the very early microbiome. It might be that they're consuming different slices of this pool. Of course, if it's B. infantis, it eats the whole pool, even though it does prefer some first and some later. B. bifidum is the one oddball here in that it consumes HMOs externally. It degrades much of them externally and then takes in pieces, and I'll talk about that in a second. And if you're interested in some of the mechanisms here, there's a wonderful review by Katayama listed here. So if you have a strain that consumes these HMOs wonderfully, and we did in the form of B. longum subspecies infantis, you should be able to provide that to a baby who's getting human milk, and it should colonize. If this is truly if HMOs are truly responsible for that colonization. And that study has been done. It was done initially by Mark Underwood in 2013, where he compared B. infantis with a strain that didn't consume HMOs at all, providing it to preemies in the NICU, and showed that the preemies that were getting human milk, and the preemies that were getting human milk that were provided B. infantis were colonized readily by the B. infantis, and the preemies that were given the B. animalis subspecies lactis did not colonize at all. This was advanced by the folks at Evolve Biosystems with Mark and Jenny Smilowitz in 2017, where they did a much more detailed study, in this case in healthy infants, and showed dramatically that if you provide B. infantis to an infant, say at day 8, you dramatically colonize their gut with B. infantis as long as that child's being breastfed. And that has major implications on the fecal biochemistries and a variety of other health biomarkers that I don't have time to get into. And I also want to give another shout out to Lindsay Hall, who just published a paper recently doing similar things with preemies. In this case, it was a supplement that had both B. bifidum and a Lactobacillus acidophilus. And this helped define that you got to have the HMO genes to colonize, because the B. bifidum colonized in the Lactobacillus acidophilus, which doesn't consume HMOs, did not. So all these studies on infants are great, and they're using whole milk, but they got whole milk. And I just told you that there's these 3 different aspects that might shape the microbiota. And so it'd be nice to try to separate this out somehow, because we don't know, was it really the antimicrobial activities that were really driving this, or was it the HMOs, the prebiotic activities driving it? And also, of course, you have a founder effect in a baby with a naive gut. So we wanted to set up an experiment that just tests the prebiotic effect. And I've had the joy of working with Helen Raybould for about 15 years now at UC Davis. She's over in the vet school, gut physiologist. So we designed the following experiment. Can you take an HMO, in this case, fucosyl lactose, which I can get in large amounts now, of course, and a consuming bifidobacterial strain, and overcome colonization resistance in a wild-type mouse? I've been characterizing bifidobacterial strains for a very long time. So I have lots of different strains, and we've been screening them on different HMOs. And we have, in a sense, optimal bifidobacterial strains for just about every HMO now. So I have a whole bunch of bifidobacterial strains to test. The study, the strains look like this. The ones that we used in this study look like this. So we used a B. pseudocatenallatum MP80 and a B. longum subspecies longum SC596. a B bifidum, and a pseudocatenatum. Now, the pseudocatenatum and the longum have genes necessary to transport fucosyllactose into the cell. That's this component here. A fucosidase genes to cleave the fucose away, and then even metabolism genes to metabolize the fucose. So both of these are quite similar in genetic profiling. B bifidum, of course, is a different profile. It consumes, as we say, outside of the cell. So the fucosidase is outside of the cell. Fucose gets released and lactose gets into the cell and is used for growth. Some of them have the genes necessary for doing fucose metabolism, some of them don't. And so I've been profiling these kinds of strains for a long time, looking for ones that grow very well, ones that don't grow at all. And of course, we have a negative control here in the JCM strain. So here's the experiment we tried. And this is just feeding some mice, either the MP80 strain, is the one I'm going to talk about first, with fucosyllactose, or we have a control mouse where we're just feeding fucosyllactose by itself. And the key part of this is we're feeding the probiotic for 5 days, but we're feeding the prebiotic, the HMO, for 10 days. So there's a stoppage of provision of the probiotic. And so one of the goals we have here is to try to understand, does the probiotic persist because the food is present, the 2FL is present? And if we just look at some of the qPCR data from this, so here's MP80 provided by itself. And you see it during the provision, you can actually do qPCR and see it increase in the mouse. That makes sense. You're giving it every single day. But as soon as you stop giving it, that population crashes. However, if you were giving it in concert with the HMO, in this case 2-fucosyllactose, you get a dramatic colonization of the mouse, which increases over the time when you stop providing the probiotic. And so we saw a 5 log difference in sometimes even larger of MP80 persistence in these mice. And of course, when you stop feeding the fucosyllactose, immediately this population crashes, just as you might expect with weaning. Now, this is a dose dependent response. If you give less HMO, you get less of a response. And we went to look at the other strains as well. And it turns out MP80 was the best strain. So here's the negative control strain, of course, and we didn't see any colonization of that. But when we looked at the other strains that could consume fucosyllactose, the MP80 was the most consistent. The B bifidum, which is the outside eater, we couldn't get to colonize this mouse ecosystem at all. So perhaps that particular consumption strategy is just not competitive in a wild-type mouse ecosystem. The other strain, SC596, was variable, and we still don't know why this one didn't colonize. We're doing a variety of metagenome sequence on this now, but we were most interested in the MP80 because it was the most consistent in this mouse model. So the next question we wanted to ask is, well, okay, so you got a way to temporarily colonize a mouse with a high amount of bifidobacteria. And I don't have time to show the stack plots in a lot of the microbiome analysis, but it covers at least 60% of the microbiota, if not more, depending on the particular mouse you're looking at in terms of the level of colonization that we would see. So the question is, if you colonize that high on a temporary basis, are you protecting the mouse in any way? Is there some benefit to the health of the mouse? So we used a DSS, a dextran sodium sulfate model. And many of you, of course, are familiar with this model. This is where DSS-induced colitis causes disrupt mucosal architecture, barrier functions disrupted, a variety of pro-inflammatory cytokines, et cetera, are induced. And it's all very noticeable. One of the things that happens in DSS models is the mice lose weight. It's one of the characteristic treatments. So we did this study here where we either were providing MP80, 2FL, in this case we provided 2FL throughout, and or DSS. And so we ended up with a variety of different treatments, DSS by itself, of course, a control treatment, MP80 in DSS, 2FL in DSS, or the symbiotic in DSS. One of the things we noticed right away is that the combined treatment, the MP80 plus 2FL, resulted in recovery of, in a sense, the weight loss in the mice, bringing it closer to normal, to the control, than any of the other treatments. And that was a really dramatic difference between these treatments. Of course, fucosyl lactose by itself also does have a beneficial effect as well. The histology of this confirmed this analysis, and you can see the control mice intestine by comparison to the DSS, which has a variety of aberrant crypts and immune cell infiltration, and pretty much the individual MP80 or 2FL mice look somewhat similar to the DSS, but you start looking at a more normal architecture when you provided the MP80 and 2FL, and that's represented by the histology scores. We knew that barrier function was also implicated in DSS, and by measuring lipopolysaccharide binding protein, we noticed that that gave the symbiotic combination gave the best analogy to the control the control treatments, and even colon length was also brought back up by both the 2FL treatment and the symbiotic treatment. We then went and looked at various inflammatory markers. This is all work done by Amy and Britta in Helen's lab. The inflammatory markers in the colon and the jejunum of these mice, and DSS, of course, has a pretty dramatic effect on some of these markers, increasing, of course, the inflammatory markers and decreasing the anti-inflammatory markers. And pre-treatment with MP80 plus 2FL reverses most of that somewhat dramatically. We looked, this is just in the intestine, but we also looked in the liver and saw similar treatments. And since we saw a systemic effect here in the liver, we went and looked in the brain and saw reversal of induced expression of various markers in the brain as well, suggesting this really truly is a systemic effect. So we believe that we can drive colonization of specific bifidobacteria with specific HMOs and come up with a model that might mimic what's going on in a baby, at least a portion of, reductionist version of that. And that has an impact on barrier function and systemic inflammatory phenotype associated with DSS. But I go back to my 3 buckets and say, wait a minute, there's still a whole bunch of activities. Milk combines these activities together. So what happens when you start combining these activities together? Do you actually see better colonization and persistence and health output? And I'll give you a little bit of that from some of the work that Vanessa did again. This is with SIG-A. So this is the work I presented before, where just pre-binding secretory IgA to the B infantis allowed for better persistence. Well, B infantis also grows on 2FL. It doesn't grow as well as the MP80 strain I talked about before, but it grows pretty well. And if you provide 2FL in this particular study that Vanessa did, we would see around 10 to the 5th, maybe up to 10 to the 6th levels of B infantis that were persisting. And of course, this is the data I showed you before, but what happens when you combine both together? Well, you see another increase. So maybe this is what milk is doing. Maybe milk is providing protection and food and synergizes in a way of delivering microbes to get their best persistence and efficacy in the gut. And can we use those lessons to learn anything so that we can maybe apply this in other situations? So I will end with commenting that we have new lessons to learn from milk. We always have new lessons to learn from milk, and that the glycoconjugates themselves may selectively bind specific targets and help enhance their persistence. And of course, the food, HMOs and the glycans on those glycoconjugates are specific substrates for growth. And we can use that knowledge to facilitate other types of microbial manipulation or gut microbial manipulation. And with that, I want to point out one way to imagine how we would do that will be talked about in Carlito Labria's talk a little bit later. So with that, I will thank the members of my lab who worked on this, particularly Britta Hess did a lot of the work, and Vanessa Castagna, and of course my collaborators. And the folks who have funded us throughout the years. And I'm going to end with this slide because I'm from Green Bay and this is an important weekend to me. Thank you very much.
Okay, thanks so much, David. That was it was a great talk. I think we're just about at time, so maybe, unless anyone has a burning question, I'll sort of ask you one. So I guess I was interested, when you're doing these sort of paired HMO administrations with the strain colonization, how specific is that to the strain that you're interested in, or do you see off-target effects on other members of the gut microbiota?
So we do see other bifidobacteria enriched in the mouse, and what we're not sure about is how connected that is to Bacteroides cleaving the fucose off the lactose, and then the lactose cross feeds. And so that's part of the metagenome sequence analysis that we're looking at now. We're trying to understand how much of a background of bifidobacteria that are growing up are really growing up solely on the lactose that's there. And I think people have seen that in other studies.
Okay, yeah, it looks like the questions are popping up. Well, we can do a couple. So the Joel Babdor from UCSF asks, can you comment on the fact that humans continue to drink milk as adults, and how do you think that would impact the way their microbiome, I guess, is structured? So
humans rarely drink human milk as adults that I know of. They drink bovine milk as adults, which has a lot lower amount of oligosaccharides. So from the standpoint of oligosaccharides manipulating the gut microbiota, that would be lowered dramatically, because the bovine milk has traces of oligosaccharides that are present in it. Of course, it has a variety of bioactive proteins, but of course, bovine milk is also pasteurized, which does a wonderful job of inactivating a variety of bioactive proteins. Not all, but inactivating a variety of bioactive proteins. That said, bovine milk is a wonderful source of protein and a very healthy drink, far better than many of the sodas we drink today. And since I'm the Shields Endowed Chair in Dairy Food Science, I strongly recommend it.
That's great. Okay, so yeah, we'll do one more. So Gail Morana from the Schwarzschild Lab here at UCSF is asking, is there evidence that supplementation with bifidobacterium 2FL in the DSS model stabilizes other microbes like Clostridia that induce Tregs and could, contribute to the control of inflammation?
Did you say stabilizes other microbes?
Yeah, I guess she's wondering whether or not there's, as you're impacting the bifidos, does that affect other immunomodulatory bacteria? That's a great question, and this is fresh data. This is out for review right now, so this is a model I'm eager to get in many people's hands. The key thing to this model is it's strain specific. You can't just take any 2FL growing bug and get it to work. It took us some time to find the right ones. Okay, great. Well, yeah, thanks again. And yeah, feel free to continue to answer questions in the Q&A box. So with that, I'm going to turn you, or it's my pleasure to introduce Kirsty Agard. She's coming to us from Baylor College of Medicine. And Kirsty is a physician scientist and gynecologist with expertise in the early life, including the in uterine environment. And so that includes the placental microbiome, the role of epigenetics in fetal programming and development, as well as microbiome interactions with preterm birth. And I think this is a particularly timely talk, given the recent series of articles that really came out in the Microbiome Journal that many of you might have seen, talking about the sort of debate over the nature and function of the prenatal microbiome. So looking forward to hearing her take on that debate, as well as the latest data from her lab. So welcome, Kirsty.
So it's an absolute pleasure to be amongst this crew, and hopefully I'm going to play a little bit of a pinch hit batter round, and I'm going to come in and share some of the maternal perspectives. And so I've titled this Womb with a View, in part because I think Our work, Susan Lynch's work, while has been well received, has also generated a certain amount of controversy in the low biomass. And I think it's important to think about this a little bit from a different perspective and avoid the sterility, non-sterility, because I believe that's kind of a solved question, but really focus in on this notion of the womb with a view. So, one of the key questions that we're interested in asking is, how is the fetus prepared for the metabolic demands of life outside the womb? And where does adaptation and evolution meet and diverge? Now, clearly, we and others have spent an enormous amount of time understanding how environmental exposures, be that the environment of the intrauterine environment or the extrauterine environment, alter the trajectory of how that baby will adapt to life outside the womb. And we've tied that to a number of things. We've shown that there are certain variants that can occur rarely or more commonly that can influence that adaptation profile, and also the role of environmental exposures on the developing fetal epigenome. We found that specifically histone variants have a much more trajectory type effect and can continue to influence that fetus long after it's born. We don't see quite as much with the methylome, but it was really the histone variants. And we became very interested about 10, 15 years ago now, as we were completing out the first round of the Human Microbiome Project work, in what role the microbiome would have across that. Now, suffice it to say, we've had quite a bit of discussion this morning on low protein, which is not a new evolutionary adaptation we're demanded to look at, but we haven't delved as much into the high fat or obese environment, because that's relatively new in evolutionary scale. And so that became something we were very interested in. Something that's not entirely separate from that is, of course, antimicrobials. And I know that Marty Blazer is going to do a great job covering this later on, but I do want to mention that antibiotics are not the only antimicrobials that we encounter, including in pregnancy. Metformin's a great example of an anti-metabolic agent that also has antimicrobial properties. And of course, timing of delivery influences this as well. It's clear that there's an impact of maternal exposures on the offspring microbiome. The question is really, when and how does this occur? So Ruth gave some great examples of where we're seeing these maternal strains, these vertically transmitted strains, as well as some paternal, that have these lasting impacts. Now, some have suggested this could simply be active or passive transfer of metabolites, but that would explain the lifelong changes, because the metabolites are by definition short-lived. They certainly could be reproduced, but they would be relatively simple. There's also been questions around intrauterine colonization, and I will say, as I always have, I personally remain agnostic as to the intrauterine colonization question. Sue Lynch's group has certainly inspired me to be a little bit less agnostic around that, but I think there's some really important questions to still ask. But we are particularly interested in this concept of immune education and enabling differential postnatal tolerance and colonization resistance and community resilience. But regardless of kind of where these mechanisms are, how is it that they translate the view from the womb? So I'm going to delve into 3 vignettes that I hope will give us an opportunity to look at this. The first is going to be lessons in microbial recolonization and community resistance from the vagina. And this is work that has been just resubmitted in revised form, as well as some work that we're pulling together for initial submission. So early on, about 10 years ago now, we demonstrated that if you compared the vagina of women, pregnant to non-pregnant, either cross-sectionally or longitudinally, we saw diminished diversity and richness in pregnancy compared to non-pregnancy. Now, it was relatively stable in that pregnant environment, but it was less rich and less diverse compared to the non-pregnant environment. And we've spent quite a bit of time since trying to understand why this is the case. And so if we just compare in a large longitudinal cohort of well over 100 women, 3rd trimester to delivery, and in that same woman, delivery to 4 to 6 weeks postpartum, we saw a couple of ecologic truths emerge. The first is that as we initially showed and Omri Koren's lab showed since, there is relative stability. If you're dominated, for example, by Gardnerella vaginalis in the vagina, it's going to continue to be a relatively stable community as you compare the 3rd trimester till delivery. But here's where it actually becomes interesting. If we then look at what happens delivery to 4 to 6 weeks postpartum, we're not seeing that same degree of stability. We're now seeing communities have much more dynamic change, suggesting that there may be some restorative potential. Now, that's really interesting because of course the vagina has to restore itself. What are the implications for that for a developing infant? Well, maybe some of those same restorative properties, either because they're conveyed by the microbes themselves, the metabolites they produce, may exist over time with that individual, which is hinted at by both Jeff's work that he shared earlier, as well as Ruth, and David brings forward within the milk. But if we look at this from a strain perspective, what we find is that there are keystone species that are present as multiple strains, but some of those are exclusionary. And if we had to characterize this very grossly between random co-occurrence or co-exclusion and examples of negative co-occurrence or exclusion, what we see is that happens not infrequently within the vagina. In other words, we could think about the pregnancy state as being exclusive and the postpartum state as being returned to permissive. So is there really truly evidence for community restoration? So to answer this question, we took advantage of some work that I've been involved in for the better part of 2 decades now in Africa, in which we have a long-standing presence in Malawi and do some vesicovaginal fistula repair work. Just to acquaint those that may not be aware, when women have prolonged obstructive labors without access to obstetrical care, they can have fistulas that occur as a result of regular contractions pushing the baby head down into the vagina without the baby actually being delivered. And as a result of that, that fistula develops either between the vagina and the bladder, or it can develop between the vagina and the rectum. And so a vesicovaginal fistula is when there's that opening that's persistent between the vagina and the bladder, or rectovaginal fistula is when it's open with the rectum. And the only treatment for this is to come back surgically and repair it. And typically we do a delayed surgical repair for a variety of reasons, but it may be anywhere from months to, in the case I'll show you of these 15 subject cohort, years. So these women have had urine leaking into their vagina for months to years. And here we're looking at the vaginal introitus samples and the posterior fornix samples. And just stepping way back on this heat map, I'm showing you here word clustering based on the Euclidean distances and showing you DNM modeling. And what you can see from a gross perspective is that there is clearly different clusters that emerge out. They're different in the posterior fornix from the vaginal introitus. Now, what's interesting is if we look at this transition state over time, setting time 0 as to when we did the fistula repair, what we find is there's some interesting community dynamics that occur. And actually within days, despite having urine leaking into this vagina for years, you start to see this vaginal introitus or the posterior fornix community both restore itself. They reach stability in the span of several days, and then they maintain that stability. Now, there's still some examples in which those dominance communities will change. But in general, especially if they go to a lactobacillus, interestingly, they'll reach that stability and they won't switch back out of it. And we can show this data in a longitudinal perspective. So here I'm showing you abundance by time by community dynamics for everything that was over 0.1% abundance. And in the vaginal introitus and the posterior fornix, I'm showing you how there was the rate of change per day, a negative change rate shown in cool to blue and a hot change rate shown in the red. And what you can appreciate is as you start to go down different phylogeny, you start to see some really interesting temporal dynamics occur. We were truly impressed by the rate of restoration that was occurring in the vagina in very short periods of time. But we sought to compare this against a larger data set because one of the things we noticed was there wasn't as much lactobacillus dominated communities in the vagina of Malawian women as we would have anticipated. Now, fortunately, several years ago, it was published a large, longer-term postnatal data set in which we had quite a bit of postpartum data on the vaginal communities in Malawian women. And there is a lack of lactobacillus dominance in about 60 to 70% of all Malawian women. And so what we were seeing was this restoration to what we would anticipate across the community over time in a relatively short interval. So what does that mean to the developing fetus? Well, what it suggests is that we actually see these same community dynamics, which may be occurring within the fetus, and some of those principles set forth in the vagina, whether it's vaginal bacteria that are actually transferred to the fetus or not, are going to be some of these same community dynamics are seen over time, which is really what both Ruth and Jeff's data alluded to. What ours would suggest is that we can use this vaginal modeling as a way to think about the modeling that occurs within the fetus. So let's move on to a second vignette. So in the second vignette, let's start thinking about instead how these restorative properties can actually communicate something to us about what happens within the fetus, and really focus in on these low biomass communities and the role of the maternal diet. Now, I want to just remind folks, without going into a lot of the what I call the great sterility debate, which I'd much rather think about as a debate around what is the means of maternal fetal communication. And just remind you that within the placenta, there is this dynamic community that is composed of the maternal immune cells, the decidual cells, which are what enable that implantation to occur, and within the fetus. And the depth of invasion is shown as we go up into the uterine wall. I will say that what we have always looked at when we've looked at the placental low biomass community is this region within here. And not really looking at any terminal villus. So this is a little bit of a cellular rich environment. Now, one of the things we observed very early on is that if we very carefully coded in and controlled for the maternal diet, we were able to see a lasting impact based upon the percent fat intake in a mom's diet, shown here with the infant stool at 6 weeks of age. This was a cross-sectional study, and we wanted to look and see if that actually held true longitudinally. And so in 2017, we published our work looking at a longitudinal cohort of over 160 women, where we were naive to kind of their mode of delivery and enrolled them in the second trimester. And one of the things we found, without going into too much detail about a number of different nuances of this published work, is that if we build generalized linear model, and here I'm just showing when we actually parse it out by different genre, at a taxonomic level, introducing formula and persisting on formula, which is actually the minority of subjects in our cohorts, or if we looked at percent of dietary intake to fat, had an interesting and lasting impact out at 6 weeks of age, in which we could jack up the amount of Bacteroides if we were using formula, and we dropped it down when we had percent fat intake. And this wasn't just taxonomic differences. If we looked at a functional level, we again saw that the maternal diet, and in this case also weight gain and breastfeeding, were really good predictors as to how we would anticipate to see the variance occurring over time. And in fact, in this cohort, we did not see a lasting impact at 6 weeks to age based upon mode of delivery. Those lasting impacts were really much more predicted upon the maternal weight gain, as well as whether or not she intaken a high-fat diet during her pregnancy. Now, we wanted to look and see if there was orthogonal evidence if we looked in other species. And for many years, we've developed a non-human primate model and utilized it to explore the epigenomic changes in response to a maternal high-fat diet. For the reasons I talked about earlier, being interested in this more novel on an evolutionary scale stress introduced into pregnancy. And what we found when we look at this cohort is that the weightedness of the effect on the developing offspring doesn't track with maternal obesity, but actually tracks with maternal high-fat dietary intake. And we use a 36% fat diet to look at this in our non-human primate models. But we were also able to delve into this a little bit more and ask the question within the context of the microbiome. And so we asked 3 initial questions. The first is, could we see any evidence if we interrupted sterile pregnancies at the equivalent to a human 32-week gestation of any low biomass communities? And could we see that difference in those communities, again, during this later mid-gestation window based upon the maternal diet? And when we did those studies, we in fact found not only could we see a low biomass community present in the fetus, very similar to the work Sue Lynch so eloquently published last spring, but we could see that it was variant very early on based upon whether the mom was fed a control diet or a high-fat diet. So here I'm showing you the gestational day 130 infants, and so this would suggest this cannot be due to contaminant alone, since we see the difference based upon the maternal diet itself, whether it was high-fat or control. And in fact, if we look at this over time, and here I'm showing you a modified Manhattan plot to tell you what of these are actually influencing at 1 year of age or at 3 years of age, where the significance line is denoted here, we do see a number of largely Clostridia and Lactospiraceae species that seem to have a very unique impact of the mom's diet based upon their relative abundance, be it up or down. And we know that this is not correctable postnatally. We published that initially in 2014 and most recently late last year. And that if we come in with a symbiotic preparation in the post-weaning interval, we can get some temporary changes, but if we stop that symbiotic preparation, we don't see a lasting impact in these non-human primate offspring microbiome. And that persistent effect of the maternal diet continues over time. And so I'm going to close with a really interesting vignette, including some data that we're continuing to work on with both David and Carlito that we're really excited about, where we're looking at common metabolites that may be important for governing these low biomass and early community establishment. And we were really inspired in this work, not only by Jeff's work from 2 years ago, but also by some really eloquent studies that David just shared with you. So one of the first things we did in a paper that was published a week or 2 ago now is we took 2 cohorts of women and we controlled exclusively what they were eating in our nutrition research center and collected their pumped breast milk. And we looked at the impact if we were giving them different sources of their carbohydrates coming in galactose or glucose, or different sources of their energy in carbohydrate or fat. And we were able to change with a high-fat diet when we just look at the milk, the caloric content of the milk as well as the percent fat, but there was really nothing grossly macromolecularly different between the galactose or glucose changes. But that was not true for the human milk oligosaccharides. If we looked at whole cohorts of either sialylated or fucosylated HMOs, we saw some specificity. We saw some specificity both within what different energy or carbohydrate sources we're doing, where we would see specificity in terms of the HMO-bound fucose versus the sialic acid based upon whether we had influenced the energy or the carbohydrate source. We were then able to correlate this with changes within the metagenomic assembly. So getting to David's point, you have to actually have the microbes present that can do something with these HMO speciations. And so we see this very interesting connection where the maternal diet is influencing the HMO profiles, which in turn influence the metagenomic, or those two could be reversed in their associations, but we believe, as David suggested, that it's going through the HMOs. We're able to recapitulate this again using some orthogonal experimentation and looking at a number of different strains that we had back in our freezers, species with fucosidase or without fucosidase, and show that we confer a different carrying capacity by bringing in different HMO preparations, and those would vary by their abundance in the milk. And I also want to just bring home this point that was brought up earlier around looking within things beyond just bifido, and very specifically within different strep and staph species as their prevalence within the milk varies. So these HMOs are such great commensal bacterial substrates. Why are they actually only found in milk? Or is that actually true? So in order to answer this question recently, and some work we're really excited about, we looked at a large cohort in this situation, 731 different women that we had done amniocenteses on for clinical indications in their mid-gestation of pregnancy. So somewhere between 14 and 29 weeks. The overwhelming majority was 17 weeks. It was rare that we were going as late as 29 weeks. And then they would go on and deliver weeks to months later. So anything that we detected within this sterile procedure had to have been in the amniotic fluid at the time we did the amniocentesis based upon their longer term outcomes. Now, we were initially doing this study for a different reason. We were doing a metabolomics-based discovery looking for biomarkers of preterm birth. And we had to set kind of a normalization standard. And so we were starting to use a spike standard that had a retention time that was very characteristic, that was present in all of our samples. And we actually hadn't looked at exactly what this was, but we were using the area under the curve of it to standardize our samples. At one point, I had mentioned to the postdoc working on this, hey, let's really look back and make sure we know exactly what it is we're using as our normalization standard. And lo and behold, it was fucosyl-lactose, which was interesting to us because we weren't anticipating to find a human milk oligosaccharide in the amniotic fluid in a collection of women whose amniotic fluid we had garnished at 17 weeks. And what became really interesting, so we dived down a little bit deeper, and it appeared to be something that was not predictive of ultimately whether they had a preterm birth or didn't have a preterm birth, but was a little bit predictive over time as to where they, the gestational age in which we collected. We did look back and have moved on now to a cohort of just 279 and done a much deeper dive and can confirm it's 3SL within a log of what we'd anticipate to see in breast milk, and using completely orthogonal methods in a really renowned second lab. We're validating all remaining 672 samples for multiple different human milk oligosaccharides. So stay tuned, we're excited about this work. Now, I want to just mention that one of the links we had established within the breast milk work is that there was a relationship between the relative abundance, at least at a metagenomic gene carriage pattern, of HMOs to the microbes. And so we're curious, since milk is also a low biomass community, would that translate into amniotic fluid? And so here I'm showing you ASV data in which we were able to use decontam pipelines and come up with a group of relatively confident calls. Amniotic fluid is similarly exceedingly low biomass, but we do believe above contaminant control levels, we do see a few microbes. And I will just say that when we bring these back against our neonatal characterizations, we do see some things that overlap between amniotic fluid and what that neonatal have at the time of birth. Now, I just want to remind folks that may not think about amniotic fluid every single day, that amniotic fluid in the mid-gestation is produced by the fetal urine. Prior to 17 weeks, it's actually placental transudate. And so we are capturing these amniotic fluids in this window between when it's coming from the fetus and when it's actually coming from the placental transudate. And what this sets up for us is a very interesting linear thought process around lower to higher low biomass community in terms of their biomass, their abundance, and their diversity. And I want to again emphasize, we have always said these are exceedingly low biomass communities, and we remain agnostic as to their colonization or not. So what I've shared with you today is some notions around the importance of a womb with a view. How it is that that mom can prepare her fetus to live outside the womb in what will need to be an adaptive environment, and the role of the microbiome in at least translating when that exposure of that mom is a high-fat diet. And we're seeing that occur not only intrauterine, but also with respect to that reinforcement in a second low biomass sphere, the milk. And so I give great credit to an amazing lab that I have. I am fortunate every day, but especially in the last 9 months, as my time has been demanded in quite a few different ways, to have such a cadre of individuals that not only care about science and the integrity of science, but each other very much. And I just am so fortunate. And we are always thankful to our funders, specifically the National Institutes of Health. And with that, I look forward to your questions.
Okay, thanks so much, Kirsty. I think we have time for 1 or 2 questions if people want to type them into the box. We have one from Elza, formerly of the Lunch Lab, who you know. I'll try to summarize. So she is asking about or she's been thinking about community collapse that occurs in the vaginal microbiome during pregnancy. She views that as a critical selection time point for microbes that could colonize the offspring.
Yeah.
But, you know, would it make sense that we have mechanisms to limit the bacteria that can be transmitted at that time point?
Yeah, so I think that's a really great question. So I absolutely that's really why we were interested in seeing, could we come up with some restorative and resilience models within the vagina to try and understand that a little bit more. And the challenge we have is we really don't see great overlap between vaginal microbes and anything that persists in that fetal, certainly not the fetal gut, and as well as neither in the fetal oral community. And that's probably not surprising because vaginal microbes aren't really meant to live in the gut. But I think the principle around how those resilience communities occur are going to be important. In addition, some of those lactobacillus species that are specifically in the posterior fornix of the vagina, and the posterior fornix is up in the top of the vagina, right underneath where the cervix is, are shared in the uterine microbiome within that deciduum, which is where the placenta implants. And so we have always been intrigued by where we see some of those lactobacilli community sharedness. And we were particularly notable that it seemed like within our restorative experiments, as well as the postnatal data, that once you hit that lactobacillus iners being a dominated community, that's where you really saw some great stability occurring. And so we wonder if part of the importance of those lactobacillus species in these low biomass communities may be to help reach that stability relatively early. So I think that's a really key component. And you are such a brilliant young researcher, Elsa. I can't wait to see what you do with that.
Excellent. Okay, well, with that, I think we're going to close the morning session soon. Thank you, everyone, for being with us. It's been a really exciting first half of the symposium. Well, we'll be back in an hour for all of the participants with our afternoon session. And so hopefully I'll see you there. For those of you that signed up for the lunch breakout rooms, I will see you in 15 minutes at the other Zoom link. So thanks again for joining us this morning, all of the speakers and the attendees, and we look forward to the rest of the meeting. Okay, thanks again.
Hello, everyone. Welcome back. My name's Katie Pollard, and I'm one of the members of the Center's leadership team. I'd like to introduce our next speaker, Nandita Garud, PhD. It is sort of a mentor's dream come true to have the opportunity in a prestigious event like this to introduce a past member of the lab. So I feel like it's a really special day for me. So for those of you who don't know Nandita Garud, she's an assistant professor in the Ecology and Evolutionary Biology Department at UCLA. She completed her postdoctoral work at UCSF and the Gladstone Institutes in my lab, where she studied the evolution of bacteria in the human microbiome. Before that, she trained in population genetics using model organisms at both Cornell and then Stanford. And so she brought that deep expertise in how genomes evolve and how alleles segregate in populations, and is one of the people who's leading the effort to take these ideas and figure out how to translate them to the complex communities in the human body. Her lab focuses on quantifying both the mode and the tempo of evolution in natural populations, with this emphasis on the microbiome. Her team specialized in the development and application of new statistical methods to genomic data, and their overall goal is frequently to make some sort of population genetic inferences. So today we will hear about that in her talk entitled Evolution in the Human Microbiome. Welcome, Nandita.
Thank you. That was a very generous introduction. I'm going to share my screen now. All right, can you see that?
Looks good.
All right. Thank you again for this invitation. It's a very prestigious group to be amongst, and really glad to be here for this event. As Katie mentioned, I've been working on studying evolution in the human gut microbiome. And so much of the work today I'll share with you is actually co-authored with her. But I will also be very excited to share with you some of the new directions that my group is going in towards the end of my talk. So the human microbiome, as you all likely know, is a really exciting area to study because it is home to a tremendous amount of variation. And this variation has been extensively studied at the species level. But what we're starting to appreciate is that there's a lot of variation also within species. And this SNP and gene level variation is actually quite tremendous. And especially so when we look across human hosts for any given species, we'll see that the B. vulgatus in me differs from the B. vulgatus in you 1 out of every 100 SNPs. That's just a lot of variation. And this variation even exists inside of a host over time. As we saw, for example, this morning in talks like Ruth Lay's talk, there can be strain replacements and so on. And as I'll show, this variation can also arise from evolution. And this genetic variation in the microbiome is actually really important for conferring phenotypes. So genes in our microbiome are associated with traits in humans like the ability to digest food, antibiotic resistance, and also drug metabolism. Now, what I'm really interested in is how is this genetic variation changing with time? And how do these genetic changes ultimately impact phenotype? And the focus of my talk primarily is actually going to be on how this genetic variation is changing with time. But I wanted to put out this question for thought, because I think that this is really important for our field. As we start to unpack the mechanisms by which the microbiome impacts our phenotypes, we cannot ignore the tremendous amount of variation that's within species. And yet, this is actually a very highly complex and multidimensional issue because you have millions and millions of features that you have to sift through. And so I think that by focusing on the evolutionary dynamics of the microbiome, we might be able to start to identify what features of the genomes inside of us are important for phenotypes. Okay, so I'm going to start off by talking primarily about how the genetic variation is changing with time. And when I talk about time, I'm talking about really different time scales, ranging from short on the range of days to months to our lifetimes to human generations. And evolution is not a static force, or it's not a constant force. It can actually be really dynamic. And what we see happening on short time scales might be quite different from what happens over longer time scales. And so we should keep that in mind when we start to think about evolution in the microbiome, because it can actually, whatever is happening inside of us may not be what is happening across the broader meta population. And I'm going to touch on all of these timescales today to varying extents. First, I'll actually speak about months, just because that's where much of our work has been so far. So when we think about adaptation of bacteria, we can look towards experimental evolution in test tubes. because we've had actually really sophisticated and detailed experiments and studies being done of these microbes in the lab. And in simple communities in which, for example, E. coli has been cultured in isolation of other species, we see that adaptation can be really rapid and common. But this question of rapid adaptation it's unclear how it translates to that of the human gut microbiome, where you have all these different hundreds of species interacting and moreover strains. Instead of seeing genetic adaptations, you might see species fluctuations in response to a selective pressure. Or you might see invasion of a better fit strain from the outside environment. Or you might actually see genetic adaptation where some modification arises de novo or by recombination and then propagates through its subpopulation in the gut. When I talk about evolution, I want to just be more concrete and say I'm talking about changes in allele frequency and not changes in species composition. And there's actually abundant opportunities for adaptation, given that it's been estimated that almost 1 billion mutations enter our microbiomes daily. And yet we have very little understanding of these dynamics inside of us. And so now I'm going to start by sharing with you work on the Human Microbiome Project data, in which data was collected on 6-month timescales, and understand what are the dynamics of evolution inside of us on half-year to 1-year timescales. And again, to refresh you, we just discussed 3 possible mechanisms by which the microbiome could respond to a selective pressure. And here, specifically, what we're going to do is focus on evolution, where we have genetic changes arising that future genetic changes can propagate on top of, on the same genetic background. I'll highlight that this work was a co-first author work with Benjamin Good and also senior authors Katie Pollard and Oscar Halachek. So traditionally, the microbiome has been studied by sequencing the 16S region of genomes. And this is really fantastic for looking at species fluctuations. However, it misses much of the relevant information that we need in the rest of the genome to study evolution. And so instead, what we wanted to leverage in our work was whole genome shotgun sequencing data, because it allows us to sample the rest of the genome. And so we can take all the DNA and shear it, and we'll get these fragments. And that's great kind of data for studying evolution and also invasions. So ideally, what we want to do with this kind of data is look for mutations that go from low to high frequency with time. And so to do that, we want to simplify the data and measure within species genetic variation. Now, what we did was we actually mapped reads to reference genomes and then identified polymorphisms within each genome using a reference-based approach. And the software we used actually is a software developed in Katie's lab called Midas. And what we want to do once we map these reads and call SNPs within each species for a given host is over time identify mutations that go from low to high. And this ideally would be a great signature for evolution, but it's not as simple as that, because multiple strains may be colonizing a host at high frequency. And so in this cartoon, we have a red strain and a blue strain belonging to the same species group, and they are diverged at this particular position where we have the mutation. And their fluctuation can then result in SNP changes that could be confounded with evolution. Now, to kind of highlight more why this is an issue, so when we talk about the strains that are present in different hosts, let's say for Bacteroides vulgatus, they're on average diverged by 1%, which means that 1 out of 100 base pairs is different. That's a lot of SNP changes to expect if you have a strain fluctuation inside of you. Whereas if you have an evolutionary event on the matter of 6-month timescales, you should expect only a handful of differences. And so those handfuls of differences could be really hard to distinguish from the thousands of differences that could accumulate over longer timescales that are being introduced through strain fluctuations. And so that's why it's really important to be able to distinguish these two scenarios. Now, the way we did that was to actually try to infer what the underlying strain structure is inside of hosts so that we could confidently track evolution. Well, the challenge is that all we witness are actually these frequencies. And so somehow from these frequencies, we have to infer the strain distribution. And so what we did was we plotted those frequencies of alleles. And these are examples from 3 individuals from the Human Microbiome Project for the species Bacteroides vulgatus. And so what we see actually is these really interesting peaked distributions, and they're quite distinct looking. And basically what they point to is actually underlying population substructure that we can control for before tracking evolution. And so, for example, in this host, we have a peak at 15%, which indicates actually that there's a strain present at 15% and another at 85%. And there were mutations that differentiate these that manifest themselves as polymorphisms here. And if you take a look at a pileup of reads, you'll see that the majority of sites belong to, let's say, the red strain. And you can kind of make the same inference for this host where you have roughly 50-50 frequency of strains. Now, as shown in these cartoons, you want to try to assign reads to one strain or the other, or in other words, phase the genomes of one strain or the other so that you can track evolution in that phased genome. And here, it's easy to see that you can assign the major allele to the major strain, but here it's easy to see that you cannot. You can actually have a lot of confusion. And so what we wanted to do was to home in on those samples that had particularly simple substructures so that we could track evolution. And it turns out that actually, when we look at those special samples that have a simpler lineage structure, colored in dark blue, we had actually quite a lot of data to work with. And so here, shown for the top 30-ish species in our data set, most prevalent species, we had almost 801 resident populations across hosts and species. And so that's actually really unparalleled data, at least in the population genetics world, to do some really nice in-depth analyses across species and cross-replicate hosts. So going forward, I'm going to actually aggregate data across these different species and hosts to make general inferences about evolution on 6-month timescales. And again, now that we've resolved the strain structure, we can ask, do bacteria evolve in the host gut? By asking, are there mutations that accumulate over time? And so what I'm going to plot is a survival curve looking at the fraction of temporal comparisons with N or more changes. And so on the y-axis, I have the fraction of comparisons with greater than or equal to N changes, and on the x-axis is the number of SNP changes. And this is what our distribution looks like. So let's unpack this. First, there's actually the vast majority of our samples, 88%, had 0 changes. And that isn't anything new in the sense that Schleissnig et al. And Voigt et al., for example, saw that in general, our microbiomes are fairly constant over time genetically. But what was new is that we saw in some percentage of our populations, 9%, a handful of changes on the order of 5 or 10 accumulating. And that would have been really difficult to observe had we not phased the genomes of the common strains in our sample. And then we also observed that in a small percentage, 3%, that we had on the order of 10,000 SNP changes. And so this bimodal distribution seemed to suggest that we have 2 processes occurring. One that generates small numbers of SNP changes and one that generates large numbers. And it turns out that those 2 processes are driven by invasion and evolution. So going back to this tree again, evolution should generate small numbers of changes, but invasion should generate a large number of changes due to the long evolutionary time that has passed. And indeed, when we look at the number of SNP differences over time within hosts, on average, we have about a few hundred, whereas between hosts, on average, is about 10,000. And so adding a line to this plot now for what's going on between hosts, we see that on average, there's about 10,000 SNP differences between hosts. And that can be used as a control to dissect what's happening within hosts. And so we see that those with small numbers of changes are experiencing evolution, and those with large numbers of changes are experiencing invasions. This was really exciting. It was one of the first empirical observations that the bacteria DNA inside of us is changing with time, and that too, seemingly short periods of time. And this isn't to be taken for granted, given that bacteria inside of us have been coexisting with us for millions of years. And maybe they could have already been optimally adapted. But no, actually, these are changing with time. And that provides a very exciting premise to start looking at the connections of our microbial genomes to phenotypes. So I just spoke about what's happening on timescales of months. Let's take a look at shorter timescales of days. How rapidly really can our microbiomes change? And so this is a collaborative effort with Morteza Rudgar and Benjamin Good, in which we looked at rapid evolution over 19 time points from a single individual for about 6 months. And these are example relative abundance distributions for 6 species. And you can see that the species distribution often is fairly uniform. In red is shown antibiotics, but in a couple of cases we see perturbations. Now, if we take a look at what's happening under the hood, inside of species, you can see that actually allele frequencies seem to change much more dynamically than what's going on at the species level. And in fact, soon after applying antibiotics to perturb the system, you can see really rapid changes in allele frequencies. And so it does seem that actually the genetics of our microbiomes are changing really rapidly on just a matter of a few days, not just months. So now let's take a look at what's going on over our lifetimes. So we noticed that there were some strains that got lost in a matter of 6 months and replaced. And so the question is, if you wait long enough, do all of the strains inside of you get replaced after 20 years? Or is it that some hosts are extra susceptible because maybe they take antibiotics? Or is it that strains continually to become so well adapted to us that replacements become less and less likely with age? And we wanted to understand the consequences of what's happening inside of us on 6-month timescales in terms of our lifetimes. But the ideal data to answer these questions really is longitudinal samples collected over a few decades. And unfortunately, that's very difficult to collect, though, again, Ruth-Leigh showed us that maybe that isn't so difficult. So what we did was we leveraged adult twins to test for long-term dynamics. And so Corpella et al. Showed that cohabiting teenage twins and siblings share many strains. And we had access to a publicly available data set by Shi et al., in which 125 adult twins were sequenced, and they grew up in the same household till age 20, and then left home and lived apart for 40 years. And what we wanted to know is, after those 40 years of living apart, do the adult twins share the same strains? If they do, then it means that the strains that are inside of you today will likely be there 40 years from now. But if they don't, then that means that everything is replaced. And so we added to this plot another line for the average number of SNP differences between twins. And what we see is that this line reflects really closely what's going on between hosts that are not related at all. So on average, twins have about 10,000 SNP changes, and that number is actually too great to be expected under 40 years. In 40 years, you'd expect on the order of about 1,000 at most. And so this suggests actually that replacements dominate over our lifetimes, even while they're really rare on 6-month timescales. Now, why these replacements happen and how they happen, or the rate at which they happen, are questions that I think the field is starting to answer. And again, Ruth Lay showed us some really intriguing data this morning addressing some of these issues. So now that we've looked at what's going on over our lifetimes, let's think about really long time scales, like over multiple human generations. And the really nice thing about the human microbiome is that we have this kind of meta population structure in which we have multiple hosts that we can sample. And the differences that we observe between hosts actually reflect really long-term evolutionary dynamics. And so it's not really clear that actually what's going on inside of us on these shorter time scales, whether those adaptations are actually adaptive on longer time scales. So for example, let's say you have an adaptation due to antibiotics, that might actually be maladaptive for transmission to another host. And so what might arise inside of you might actually get purged later on. But we wanted to go in and take a look and see whether those adaptations inside of you are adaptive on longer time scales. And so just some theory to put some framework behind this: adaptations persist, but deleterious variants will be expected to be purged. And so I'm coloring on this tree non-synonymous mutations that may or may not be adaptive, but more likely deleterious, and synonymous mutations. And if they're maladaptive, or then they'll disappear with time, and you'll have a low non-synonymous to synonymous ratio, indicating a lot of purifying selection has been acting. But if you have less purifying selection, maybe because some of these are adaptive, you will have fewer disappearing with time. And so what we did was we plotted the ratio of DNDS against the divergence between strains found in different hosts. And this is a plot recreated from original PLOS Bio paper. And you can see that actually on average, DNDS is on the order of 0.1, indicating that purifying selection dominates over long timescales. One of the cool things I'll highlight about studying the microbiome, at least from a pop-gen perspective, is that we can start to dissect differences across species. And you can see that although one model seemingly fits all species, there are important differences to dissect. And we can start to dissect some of these by looking at different pathways. And so here, I've not enumerated them just because the font gets really tiny. But in each black dot, we're looking at the mean DNDS for different KEGG pathways, and each colored dot represents the DNDS for a given species. So the mean is for a given pathway across the genes in that pathway for all the species aggregated. And what you see is that there's a really wide range going from less than 0.1 all the way up to 0.5, never really surpassing 1, suggesting that there's not really any evidence using this statistic, at least, for positive selection. But there is evidence that there are different pathways experiencing different levels of constraint. And that, again, could be really useful information for tying back the genetic variants in our microbiomes to phenotypes, or in other words, assigning functional importance. And I'll highlight that this is work in prep with my postdoc, Will Shoemaker, and my undergraduate student, Daisy Chen. We also are finding a really interesting relationship between the variance of DNDS and the mean of DNDS. And this positive correlation makes a lot of sense, given that if you have relaxed purifying selection, you can have more room to wiggle. And so this might also be another hallmark for relaxed purifying selection. Finally, I have shared with you different timescales on which our microbiomes can evolve. But now let's talk really briefly about how do these genetic changes impact phenotypes. Ultimately, that is a great mission that I think we want to pursue more broadly in our field. And I don't have the answers to this question. I think this is going to be a huge effort going forward. But in my group, what we've been looking at is how, in general, can we make robust associations between microbiome composition, either measured using OTUs or K-mers, with phenotype. And I'll highlight a brief case study on colorectal cancer. So to make good inferences, we want to have large sample sizes because that gives us more power. But the problem is that a lot of data sets are actually fairly small. And so people are starting to stitch together data sets because they measure the same phenotype. And multiple groups, for example, have measured colorectal cancer. When you do this, you actually can get a lot of confounding covariates arising from these stitched data sets. And here I'm showing that those data sets popping out in a PCA plot when you plot the first two PCs. And this isn't just specific to stitched data sets. In the American Gut Project, for example, we see a lot of confounding covariates as well. And surprisingly, when we plot color the dots by case versus control, you don't see such a clean separation of points as compared to when you color it by data set. And so really these confounding covariates can dominate the variance in your data sets. And this can be a really big problem going forward and when you want to make robust associations with phenotypes. So there are fortunately some methods for correcting these covariates like COMBAT, LIMA, and BMC, batch mean centering. However, we've been trying to look into even better ways to deal with this covariate correction method, and really proud to share that my graduate student, Leah Briscoe, has been working on this and finds that regressing out top PCs can actually be fairly effective. I just wanted to note that this is from looking at k-mers in microbiome data, which may be a hair closer towards SNP level variation rather than OTOs. But we see similar effects also with OTOs. So
Nandita, I think you're close to wrapping up, but you've got 3 minutes.
Thank you.
Till including questions.
Almost done. So I wanted to just say that, we're I think a big challenge again for the field is to connect these genetic changes in the microbiome to phenotype. And I think that by looking at the evolutionary dynamics inside of us, we will start to be able to do that more effectively. But again, once again, the population structure and so on is going to be a huge confounding covariate to deal with. So to conclude, bacteria can evolve in the human microbiome on 6-month timescales, but replacements are rare. Over our lifetimes, bacteria are replaced to suggest limits to how well bacteria have become to us. Over generations, the adaptations that accumulate inside of us are purged, and next steps are to connect genetic changes to phenotype. I'd like to thank Ben Good, Katie Pollard, Oscar Halachek, Will Shoemaker, Daisy Chen, and Leah Briscoe. This is my lab. Please contact me if you're interested, and I'm happy to take questions.
Thanks, Nandita, for the exciting talk. We have a couple minutes. If anyone has a question, please put it in the chat. I can ask one while we wait to see if anyone else does. Nandita, it sounded like the principal components you thought were capturing things more like batch effects. Do you think they also capture the population genetic structure? And how would you know if that was true?
I think that's a really important question, and we haven't reached that point yet, but I think it's something that we need to answer. And I think that, PCA, for example, has been famously applied to human genetic data in like November et al.'s work. And it's something that will certainly need to be corrected for before we can connect microbial genotypes to human phenotypes or even microbial phenotypes.
Thanks. Anyone else in the last minute?
I think I see the questions actually.
Oh, they are in the Q&A. Got it. I thought they were in the chat. I got it. Great. Nandita from Preston Tassoff, has this approach been applied to environmental microbiomes yet?
Not as yet, but I think it would be, well, actually I should highlight Alex Kristoff has done some really nice work on the population genetics of soil bacteria, but we don't know how they evolve on short time scales.
Great. We'll do one more. Outside of antibiotics, what do you think contributes to strain variation? And how much do you think, well, let's just stop at that one because the other question's unrelated.
Sure.
So, yep.
I think it could be a range of forces, mutation that are spontaneous, recombination, again, spontaneous, other selective pressures like diet, medications, but we don't quite know what exactly is happening yet.
Awesome. Well, thank you, Nandita.
Thank you.
It's my pleasure to introduce our next speaker. Someone else who is known to our UCSF community, Mohamed Dania. Dr. Dania is Associate Professor of Molecular Biology at Princeton University. He's received multiple different awards, including the NIH Director's New Innovator Award, and he's a member of the SAB for Deep Biome Therapeutics. His research is focused on small molecule mediated interactions in complex communities and microbiomes. And he's interested specifically in their potential as therapeutic agents. His lab is working to develop the necessary computational and experimental tools, so kind of do them both, to functionally study interactions mediated by the uncultivatable members of complex microbiomes. So I'm looking forward to the talk, Mohamed. Your talk title is Production and Metabolism of Bioactive Small Molecules by the Human Microbiome.
Yeah, thank you very much, Katie, for the nice introduction, and thanks to the organizers for the invitation. Of course, it's really a pleasure to be back, virtually at least this time, at UCSF. This is my home, and I'll come back every single time I'm called. So really happy to discuss today with the students and to learn about all of the new stuff that's happening at UCSF. I did change the slides a little bit and the title, but the theme is still similar. So basically, in my lab, what we're trying to do is to understand the microbiome from a slightly different perspective. So I'm going to talk less today about disease relevance, but more about how we can look at the microbiome itself as this repertoire of biochemical potential. We always call it in my lab as this really high density, high diversity bioreactor. And the complexity here is that it varies widely between individuals as well, because the composition varies between individuals. A rough estimation tells us that there are hundreds of thousands of pathways and biochemical pathways and enzymes that are encoded in the microbiome, and we really do not know what the majority of those pathways do. And the idea is that, if we can deconvolute this enormous capacity, we'll start learning more about what the microbiome can do, and also about how it relates to our health and disease states. So today I want to tell you about a little one approach that we thought we could take to start tapping into this enormous biochemical potential. And this approach comes from studying pharmaceutical drugs and how they interact with the microbiome. We really wanted, when we started this, we didn't really focus, we didn't think too much about the pharmacology, even though I'm a pharmacist and this is something that I'm really interested in. But I really wanted to find a way that can reveal, the enormous capacity of the microbiome to interact with a large variety of compounds, of chemicals. And luckily, we are doing that every day. We're giving people drugs every day. So we went to the pharmaceutical drugs as being our probe molecules to start revealing the biochemical potential of the microbiome. And as you can see here, from just 24 or so molecules, we can see the enormous structural diversity in these pharmaceuticals that we use, both natural and synthetic. And of course, along the way, we will learn a ton about how the microbiome interacts with these drugs. How would that affect toxicity, efficacy, and bioavailability of these molecules in humans or in mice or in any other organism. So this is nothing new, and definitely not new to this audience or many others in the field that have worked on this way before us, including Peter and many others. and a few key references are mentioned here. But we wanted to take a slightly different approach towards solving this problem. And again, I'm presenting it today from the goal of finding new biochemical reactions and biochemical pathways that we can understand that are, undergone in the microbiome. So our new approach that we wanted to take is a whole community approach. So instead of thinking about bacterial cultivation and studying one species or strain at a time grown in the lab, we thought that we can take a whole community approach where we can culture an entire microbiome, or as entire as we can make it in the lab. And then this would allow us to do a few things that a single culture method cannot do. One of them is these multi-organismal contributions. So we often see that there are opposite reactions that could be catalyzed by different microbes. So let's say you have one chemical, one microbe will metabolize it into one compound, and then another microbe will actually reverse that reaction. So this would allow us to think about these net effects of, how can we study the effect of all of the microbes together on one drug or one chemical. We also understand sequential metabolism. So instead of reversing that reaction, what if a second microbe takes a step further and does an additional modification to it? Again, we also hope to study inter-individual variability. So one drawback of studying cultured isolates is that, we would work with key isolates that came from one person or another, or are available in ATCC strains, or reference genomes, or have reference genome sequence, but these may not represent the repertoire of strains that something that we've been talking about today in several of the talks that exist in people. So if we can do it in a personalized manner, culture the microbiome of each person individually, then we'll be able to understand also strain to strain variation. And then the last goal was to try and bring some global understanding of this chemical metabolism that happens in the microbiome. Because if we analyze hundreds of drugs at the same time from many different people, or against microbiomes from many different people, then we will start to draw major conclusions. And that was the goal. So I'm going to tell you about the study that was recently published, and then I'm going to end up with a couple of unpublished results. So in this study, basically, we wanted to develop a culturing method that would be appropriate for different microbiomes, and will be able to capture a lot of the diversity that exists in the microbiome, as at the strain level, or as much resolution that we can achieve using 16S sequencing. So what we've done here is that we basically collected fresh microbiome samples from 20 different donors, tried 10 different media, and then analyzed the microbiome composition using 16S in comparison to the fecal samples themselves. And then we did also biomass measurements, and we developed a small algorithm that would allow us to incorporate all of this data together and select the best media that would allow for the culturing of the majority of the microbiome from all of the donors. And this, again, takes into effect the biomass as well as the microbial composition. And this method also allows us to not only select, but to compose. So we were able to actually compose a new medium. None of the 10 was good enough to our limits, and we designed a new one that was a composition of 2 different media that achieved the best outcome. And this project was led by 2 phenomenal students in the lab, Bahar, who's an MD PhD student, and Jaime, who's a quantitative and computational biology PhD student. So this is one major result that we obtained. So it's a slightly complex figure, but basically you can see that each box here is a single donor out of the 20 that we did this approach with. Each column here is a single sample that is either, first column is the fecal sample, and then the 10 different media that we tried. Each row is a taxa that is here just colored by the order level, but it's an ASV level resolution, artificial sequence variant, which is as close as you can get to strains using 16S. And as something that you may already appreciate is that we can definitely see personalization here in the culturing method. So this stripe exists in donor 2, it does not exist in donor 1. This stripe exists in donor 1, does not exist in donor 2, and so on. So you can see that there is some level of personalization that is coming from the fecal sample and replicated in the culturing media. We were able to quantify that by basically looking at the recovery of the ASVs in the culture. So this is just the number of ASVs when we compare donor to donor, just the fecal samples of the donors, and you can see that there is definitely personalization here. This is on the donor themselves, but this personalization is also replicated if you compare the donor fecal sample to the cultures that came from that particular donor, in comparison to all of the cultures that came from many different donors, or the fecal samples from many different donors. So if you have a fecal sample from one donor, it's the closest sample that we have to it is a culture that came from that donor versus any other cultures that came from any other donor. So we can achieve that level of personalization in our culturing approach. And we can also achieve a great level of recovery that would actually surprise us. We can achieve about 80% recovery on the ASV level for anything that is 1% or higher. And that drops a little bit, when you go below 1%, but still, above 60% or so. So we're definitely recovering a lot of the organisms in the original sample, and even rare ones we're also recovering. And we're doing that on a personalized manner. So we ran with this, we picked a medium that we like, and then we screened a drug library of 575 drugs against one donor sample. We just wanted to answer one question: how much can a single microbiome do with the appropriate controls of media only, heat killed, all of that stuff. But the idea here was very simple. We incubate replicated copies of the cultured microbiome with different drugs, with each of the 575 drugs, and we do LC-MS analysis. And basically, we're very stringent at this level. We're looking for two things: either the appearance of a new metabolite. So here, drugs 1, 2, 3, after incubation with the microbiome, nothing happened to them. Drug 4, if you have an appearance of a new metabolite, then we would call that positive result, microbiome-derived metabolism positive result. Or if we see the drug completely disappearing, complete depletion, we would also call that a positive result. We did not do partial depletion here in this one assay. So we were very stringent when we started. And even though we were stringent, we were able to find 57 cases of microbiome-derived metabolism, most of which had not been determined before or reported in the literature. This accounted to about 13% of the tested drugs being metabolized to this very stringent level by a single microbiome sample. So we were very excited with this result. And we, of course, went ahead and characterized a few of those by following the metabolites, purifying them, solving their structures using NMR and mass spec. And we found really interesting modifications. Here is, for example, a deglycosylation reaction. We find common ester hydrolysis or thioester hydrolysis reactions, but we found them on new drugs in this case. We found a relatively uncommon esterification that is preceded by a nitroreduction reaction, and so on. We were able to find a lot of known and new modifications, as well as many cases of drug depletion. We went ahead and said, okay, does this happens in our ex vivo cultures? Does this occur in mice? So we picked candidates, and we wanted to follow up with a mouse PK study, pharmacokinetic study. And the way we design these studies is that we have differentially colonized mice, either without a microbiome by antibiotic treatment or germ-free, and with a donor microbiome in them. And then we would just give a single dose, a human equivalent dose, and then we would follow the drug in the blood and the feces and urine. And we were interested in this class of molecules that I just told you about. So capecitabine is an anti-cancer drug, a prodrug, and we found that it's deglycosylated into D-glycocap, which we wanted to see if that occurs in vivo. Also because this is just one member of several compounds that we found to be undergoing the same reaction from the same family of fluoropyrimidines. The design of the PK is as follows. In this case, we needed to use antibiotic treatment. So we cleared the microbiome for 2 weeks, and then we would give a single dose, colonize or not, give a single dose, and then follow, do a proper PK every 20 minutes and look in the blood and feces. And we were very pleased to see that if you have the microbiome, at least in feces, you can determine the presence of the D-glycocab, the metabolite that is microbiome dependent, while if you don't have a microbiome, basically this compound is non-existent in these mice. So this was, a nice result that what we see ex vivo, we can actually see in vivo. We also wanted to follow up on this and see if we can utilize this method to be able to measure inter-individual variability. That was one of the goals that I told you about at the beginning. So we did this in a high throughput way where we selected 23 drugs, including some that came from our original screen, and we cultured them with the 20 different donor microbiomes along with the controls, the heat killed, the medium only, and so on. And we followed that with quantitative, targeted, and untargeted metabolomics. So in this case, we were able to measure also levels of partial depletion of these drugs, of the parent drugs. So here, again, another kind of complicated heat map, but I'm going to walk you through it. So this is basically donor 1 to donor 20, and here are the 23 drugs that we tested. And what we're measuring here is the level of the drug after incubation with the microbiome from each of those donors. And if you see an asterisk there, that means that this depletion that occurred in that drug was statistically significant after comparison to a heat-killed control and a medium-only control. So you can see that, a couple of patterns here. There are some drugs where basically nothing happens. So if you see a yellow stripe like this without any asterisk, that means that that drug was rock solid. It did not get metabolized or modified at all by the microbiome. At least it didn't go in concentration, down in concentration at all. There are drugs that always get metabolized by any of the microbiomes. And then there are ones that actually vary, and those are the most exciting ones to us. They vary between donors. Some donors can do the metabolism, others cannot. And those include, for example, digoxin, which is a drug that Peter worked on extensively and showed that this level of inter-individual variability is clinically relevant and happens in people. We also can do the same type of quantification on the metabolite production, and we see exactly the same thing. We see cases where all donors produce the same metabolite. We see cases where none of the donors produce any metabolite. And we see cases where some donors produce one metabolite and other donors produce a different metabolite for that same exact drug, which is something that's super exciting for us because, again, that tells us that the biochemical capacity of an individual microbiome sample can vary widely between individuals. And this is one drug, for example, that we're following up on, nicardipine, where there were a ton of metabolites that we discovered, 12 to be exact, and their patterns varied widely between the different donors. So what does that mean clinically? We still don't know, but that's something we're very excited about, both from the biochemical basic science understanding of the enzymes that are encoded by the microbiome, but also from the clinical perspective as well. This is a simpler way of looking at the same data. These are the triplicated samples. You can see that we're quantifying the metabolite in the different donors, and you can see for some drugs, basically all donors produce the same levels of the metabolite. in other drugs, like the digoxin case that I told you about, only 3 donors can produce statistically significant levels of that metabolite. The last part that I want to talk to you about is how do we take this from, kind of a biochemical screen that allows us to identify new metabolites and known ones and to quantify them and compare them between different microbiomes to actually linking those to specific genes and microbes in the microbiome. So we took 2 separate approaches to answer this question. The first one is a homology-based approach. So just based on our biochemical knowledge, if we look at a reaction like this, again, this is the anti-cancer prodrug capacitabine that is deglycosylated into D-glycocapacitabine. And we wanted to know, okay, what enzymes could be carrying out this reaction. And immediately when we looked at this, we thought, okay, this reaction could be catalyzed by either thymidine or uridine phosphorylases that exist in many bacteria in the DNA salvage pathway. So, okay, that's exciting. We can now take a homology-based approach to search for enzymes that are from these classes and see if they perform the reaction. In order to do that, we wanted to find a cultured microbe that would be kind of our case study to see if we can use that homology-based approach to find the right enzyme. So in this case, we just screened against a very small library of gut microbes, and we were able to find that, very pleased to find that E. coli can perform this reaction, so why not start easy? We took E. coli, we looked in the genome, there are definitely 1 copy of thymidine phosphorylase encoded by DOA and uridine phosphorylase by UDP. We knocked out one or the other or both, and then carried out the same experiment in an ex vivo setting to see if the wild type or the knockout E. coli can perform the reaction, and we found that only UDP was required in this case. If you knock out UDP or both, you lose the ability to perform this particular transformation. So only UDP was involved here in cap deglycosylation. Next, we looked at the other compounds that are related to the capacitabine itself, and we were super happy to see that there is actually some level of substrate specificity here. While in the case of cap, only UDP was required to perform this reaction. In the case of these other, structurally related molecules, both UDP and DOA or thymidine and uridine phosphorylases can do that, can perform that reaction. So this is kind of an advantage that comes with the ability to screen a large drug library, because you always have derivatives of the same drug, and you can start to draw conclusions about substrate specificity. And a second approach, if we didn't have a cultured microbe that performed the reaction, and this was one of the cases in our study, we were very excited about hydrocortisone and hydrocortisone acetate being converted into 20-beta dihydrocortisone. This was not a reaction that was observed in human microbiome before. The 20-alpha was previously observed by Clostridium scindens, but not 20-beta, and these 2 steroids have different activities. So we're excited about that, and we wanted to know, with that very clear stereospecificity, which enzyme could be carrying out this reaction. We could not culture a microbe that can do the reaction, so we went ahead with a functional metagenomic approach, where we basically take the DNA from the donor fecal sample, chop it up into small pieces, clone it into expression vectors, and then transform into E. coli, pool these transformants into pools of 20 to 60,000 clones, unique clones per pool, and then screen those using our approach of ex vivo screening. So doing that, we were able to screen 3 million total clones. And here it's just, I'm showing the level of the 20-beta dihydrocortisone production by the different pools that we screened. We start with a very small production level, but our quantification was enough to pick that out as being significant. And then we were able to enrich for the clone that can perform the reaction. Out of 3 million clones, we found actually 2 at the end that can perform the reaction, and we sequenced them and found that they encode indeed encode a 20 beta hydroxysteroid dehydrogenase enzyme that was never characterized before. So we were very excited about this, and as kind of a possible approach to find new enzymes that can perform reactions that where these reactions are not nicely catalyzed by cultured microbes. And these enzymes that we find, they're not rare, they're not specific to our donors in any way. When we look for them in large metagenomic studies, we find that they exist in abundance. Here are just the quantification of the prevalence or the median abundance in RPKM, and again shown here for the enzymes that I talked to you about, the UDP, the DOA from E. coli. You can see that they can be abundant in populations, and also they vary in abundance from person to person. Same thing with the 20 beta hydroxysteroid dehydrogenase that we discovered. It's actually pretty abundant in many different populations, and again varies in abundance from person to person. While in comparison, this is the 20 alpha. It's very rare in population. The last piece that I want to tell you about today, which is an ongoing study that is unpublished, is basically, again, to look at this idea of substrate specificity. And this is related to the two anti-Parkinson's drugs that are related to each other, tolcapone and entacapone, where we can observe sequential metabolism, that starts with a reduction, a nitro reduction, where almost every isolate that we tested can perform the reaction, to an N-acetylation where very few isolates can actually perform the reaction. What was super unique about these two compounds is that we started characterizing N-acetyltransferases in the organisms that can perform the reaction. We looked, for example, at E. coli, which can perform the reaction with tolcapone, but not entacapone. We actually characterized 16 different potential N-acetyltransferases in E. coli. Only one of them can perform the reaction for tolcapone. None can perform it for entacapone. We then looked in Parabacteroides cystisonis, and we see exactly the opposite. We characterized 10 different potential N-acetyltransferases. 9 of them can perform the reaction for entacapone. Only one cannot. So this is just to tell you that, again, by looking at these enzymes a bunch at a time, we're learning a lot more about the substrate specificity of these enzymes and what they can perform reaction-wise. So I hope I told you today that looking at the microbiome as a community and as a way to perform many different reactions together is an interesting way of approaching this problem. And I'd like to thank the lab. Already thank the two people who were really important for this work and carried out all of the work. And thank our, of course, funding agencies. And thank you again for your attention and invitation.
Thank you for the exciting talk. We'll look in the Q&A for some questions. There are two here. First one is, have you monitored the drug-exposed microbiomes longitudinally to see if non-antibiotic drugs and/or their metabolites modulated the microbiome-mediated metabolism of other sequentially administered drugs?
Yeah, we've not done drugs in combination yet. We are thinking about that. If we can put drugs together with the same microbiome, what would happen? We're looking at the effect on the microbiome as well. So we're trying to see, if the drugs can also are affecting the microbiome and microbiome metabolism. But in our case, we're not, we don't expect too much signal because we incubate these drugs. We try to simulate as much as possible what's happening in the gut. So we incubate these drugs with almost a stationary phase of the microbiome culture. So these are not like super actively growing, but they're they finished growing and they're sitting there. They're still metabolically active. But these are interesting ideas.
Yeah. Do you have any thoughts on drugs administered by infusion, IV or IM?
Yeah, definitely. So many drugs can go through biliary secretion and end up in the gut. And we're thinking about that. We have not. We're trying to design a good way of approaching those. We have a couple of examples that we're working on, but nothing systematic yet.
And for drugs with partial metabolism, do you know if partial metabolism is due to kinetics or suicide intermediates?
Yeah, we don't know yet. So we think there are several ways of, why we see partial metabolism. Yeah, in some cases, it's not just the right substrate for it, and the enzymes are just very slow. We've seen that. So that's mostly enzymatic kinetics. In other cases, maybe the drugs are not actually getting into the microbes, they're not absorbed, so it's something actually limitation on the transport of the drug into the bacterium. There are several, potential explanations there.
Great. You have a couple others in the Q&A if you want to check it out, but I think we'll ask you to stop sharing your screen now so we can stay on schedule. Thank you so much.
Yeah, thank you.
I would now like to introduce Tiffany Scharschmidt, who will moderate the next couple talks.
Thanks, Katie. Thanks, Mohammed. It's I wish we were in person, as I'm sure you all do as well. It's been fun to meet new faces and hear from old friends. Mohammed and I were postdocs together. So like Katie and others, I'm joining you today as a part of the leadership committee here of the UCSF BCMM Center. And thanks to others for really leading the charge and putting today together. But it's my pleasure to introduce our next speaker, Carlito Labria. Dr. Labria is joining us from UC Davis, where he's a professor of chemistry and biochemistry. And his lab is focused on analytical chemistry with a specific emphasis in the areas of nutrition, disease markers, and bioanalytical mass spectrometry. His team is pioneering glycomic approaches for early diagnosis of cancer, and the research in nutrition, which I believe we'll hear more about today, examines human milk as a model for the perfect food, and he seeks to understand its bioactive components. So welcome, Dr. Labria, and his talk today is entitled Structural Analysis of Carbohydrates in Human Milk and Weaning Foods is Key for Understanding the Trajectory of the Human Microbiome.
Thank you very much for that kind introduction. Are you guys hearing me and seeing this fine?
Yes.
Okay. So I'm going to talk about the other end of the microbiome, and that's essentially what they eat. And so we've been looking at carbohydrates for a long time. And one of the reasons why we've been interested in it is that it's one of the most abundant biopolymer in nature. And the interaction between the most abundant biopolymer and one of the most abundant life form should take place. And so we've been developing methods to try to understand what these carbohydrates do and their structure. And of course, the plants make up a large part of that carbohydrate structure in the nature. And so one of the things that we've been doing is is now to characterize what those particular as they relate to food. But animals are also, like us, are also glycosylated. And so a lot of the plants have cell walls that are carbohydrates, but also animals themselves. And where they're found is on the surfaces of cells. And I show you this glycocalyx, because this is what bacteria would interact with in both commensal and pathogenic. And so what we do in our group is we develop methods for all kinds of carbohydrates, but also on the cell surface. And so, for example, on a surface like this, you'll see all these different types of glycosylation. And over the years, we have methods that can characterize them with deep structural detail, and we can even sort of modify them. And so this is sort of a side topic, but what it shows you is that this is this glycan, if you release them all, you get a chromatogram, an LC mass spec chromatogram that looks like this. There's about 300 to 400 structures here, but I just highlighted the big ones. And so what we can do is we could grow a cell culture, and we can actually change the glycosylation just by putting in different things. And when you do that, what you do then is you change the glycocalyx, and you present to different bacteria different types of glycosylation. And so we can do certain things. For example, this is Salmonella typhi. And what we've done is we've looked at adhesion as we've altered the glycosylation. And based on this is the one that typhi seems to prefer. And it turns out that's something that's highly fucosylated. Whereas in invasion, it turns out that it seems to prefer high mannose glycan. And so based on this interaction, we can say something about typhi, about how it infects. The other thing that we could do, this is just to show you that these are all significant. The other thing that we could do is, now that we found that, for example, fucose will block binding, we can then incubate the typhi with different amounts of fucose, and you could see that we could diminish, conserve the adherence and also the invasion properties of that compound. So again, that just sort of illustrates the role of glycosylation. But but what I'm going to focus on today is how food and how that will affect glycosylation. Food, if you think about it, is just a carbohydrate structure, just like you saw in the cell surface. And so the question then is, how do we characterize food? And that's what's actually been quite difficult. So the microbiome and the genome has got incredible changes, and now everyone can pretty much determine the genome. Carbohydrates has not been so advanced. And in fact, a lot of the methods today for carbohydrate analysis and in food require a lot more material. They're not very quantitative, and they haven't been updated really to modern instrumentation. So the methods today actually come back from the 60s. And so, as I said, genome sequencing has advanced considerably. Proteomic sequencing is the same way. On the other hand, our method today goes back to Hakamuri's method in the 60s. And so what my group's been doing is really trying to create what we call the next generation of tools to do glycomic analysis. And this requires a lot of mass spectrometry, a liquid chromatogram and mass spectrometry, which we have in our lab. And so when you know the structure of something, you can do something really important. And one of the things that we did was earlier on was characterized with great detail and with broad coverage was the structures of human milk oligosaccharides. And so people cite that there's about 200 structures, but if you dig deep down, there are about 300 to 400 structures, not all found. And we could also phenotype based on this structures. And so, for example, one phenotype would look like this, and the other phenotype would look like this. So this, I could tell right away, is a secretor phenotype because of that 1,2 fucose, whereas this one has 1,3 fucose. And so this is a non-secretor. And so what we found, actually, David Mills, who figured out this group figured out every single enzyme that breaks it down. And so there's a one-to-one correspondence between, for example, this linkage and the different enzymes that are associated with the bacteria itself. And so being able to determine the structure. And so what we did is that we actually fed these bacteria these oligosaccharides, and we can watch them. We can watch these things disappear. We can monitor them quantitatively, and we could tell you the actions of the enzymes that are available there, not just for this bacteria, but for any bacteria. And over time, you collect a bunch of human milk, and so we put those together in this large chart, although about over 1,000 samples. And what we found is that the secretors are about 20%. People say that at the time, all the time, but that's really refers to European and also Americans. But in West Africa, there's about almost 40% non-secretors, the same way in South Africa. In India, there's a lot of non-secretors, and you go out towards Asia, East Asia, then there's, again, fewer non-secretors. And then you go all the way out, the out of Africa model, if you go out to indigenous tribes in Bolivia and Peru, this is Katie Heinz work, we find that there are almost no non-secretors as you go out to South America. And so again, if you know structures, you can do big things. And one of those is identifying this bacteria. And of course, now we have a product Evolved Biosystem sells this, and it's now accepted in hospitals like Kaiser, Oregon, and so on. And so mammals, we've always thought, has this really great strategy for attracting microbes, right? You feed it, and we thought, wow, that's pretty impressive. We mammals are so cool. Well, we got involved in this project with Alan Bennett and the Mars Corporation, and what they wanted to know was that there was this plant. It's a corn, but the roots here secrete this mucins, and they want to. And this mucins, it turns out, attracts rhizobia bacteria, which fixes nitrogen and fertilizes the mucins. And so the question was, what is the structure of the mucin? So my group did, over a period of a year, did a de novo sequencing of this structure. And what we found was that it looks like this. So this is based on a lot of work, a lot of LCMS, fragmentation, dissociation, and so on. And if you look at it, the composition of this compound, it contains fucose. So it's one of the biggest fucose-containing compounds that we observe outside of seaweed, of course. And so that's sort of fascinating that between humans and mammals, between animals and plants, that they would use sort of similar compositions of compounds. Okay, so now we know that structures of food are important, but we've been limited by the methods that can actually structurally characterize food. And so we like to think of food as really a post-genome product, mainly because you can have all these different species, all the single species of food can give you really totally different food, right? And this is just the eggplant. If you can imagine how many different eggplants there are, so the genome's the same, but what's different about this is really the metabolites, the carbohydrates that are produced. And so the question then is, so now we don't just need to figure out what the genome is. We need to figure out what the glycome is and the carbohydrate that makes up all these different plants. And so again, as sort of our human arrogance, we think, wow, human milk is really hard to do. And in fact, it was difficult because I always like to point out that it took about 80 years to figure out that they were food for the microbes. But if you look at the potato, the humble potato is actually a lot more complicated than human milk. And so then that's sort of fascinating, but at the same time, these structures have monosaccharide compositions, have linkages just like human milk. And so there must be a microbiome associated with the potato. And for that matter, there must be a microbiome associated with any kinds of food that you perhaps grew up with. And so the problem with carbohydrates is that not only are the structures important, but the degree of polymerization is also important. So monosaccharides themselves, glucose, galactose, fucose, sialic acid, have their own biological properties. But if you put them together as oligosaccharides, they maintain some of those properties, but include some of their own. And then when you make long polymer chains, they get yet new properties. And so what you need to be able to do is figure out all the different structures when you have carbohydrates. And that's been really the big issue, because you don't have this in the same way as you would with other biopolymers. And so when what we've done, and what my group has done over the years, is really come up with methods that, for example, can characterize over 300 structures of free oligosaccharides in human milk, look at the N-glycans on cell lines like I showed you, the O-glycans, look at glycolipids, and then we've looked at all the glycoprotein permutations of these things. The last challenge to us was the polysaccharide, and how do you figure out those structures? Well, if you think about a polysaccharide, it's made up of monosaccharides, and it's also made up of specific linkages. And so to a first approximation, getting a monosaccharide composition, we thought would be a great way to characterize it. But however, if you can get the linkage information, that would also be really useful, because again, there's specific enzymes that break down very specific linkages. A 1,4-glucose, for example, will require a different enzyme compared to a 1,3-glucose compared to a 1,6-glucose. And so being able to eat these linkages and the monosaccharide composition would be really useful. And so what we did was we developed a bunch of new methods to make them rapid throughput. And also very sensitive. And so this is what it looks like to a chemist. This is very interesting. To anyone else, probably not. But anyway, what you get in the end is a chromatogram of all the different monosaccharides that make up your compound. This requires a lot of mass spectrometry. In this case, we use a triple quad using multiple reaction monitoring. You have to find the right separation method. This is in a 96-well plate format. But in any case, this whole run takes less than 10 minutes, but the sample prep takes time. But what you get in the end is you get about 15 to 20 monosaccharides that you can monitor simultaneously. And so just to put this place in context, this is the current method. So to do, let's say, hypothetically, if you were to do a 96 samples, this would take 53 hours. Ours would take 3.5 hours. This would take micromoles, ours would take femtomoles, and so on. And you can ask, well, there's a lot of food. Why do you need the sensitivity? Well, if you're doing clinical trials, and if you want to look at feces or urine, you need that kind of sensitivity if you're trying to monitor where these things go. So when you have a fast method, then you can start looking at food in general and looking at food as in a new way. So this is a survey of about 1,500 foods based on their monosaccharide composition. And if you were to blow up a strip of this, this is what it would look like. And so what we would have is glucose, and then all these other monosaccharides that make up that food. And so this is just ordered from low glucose to high glucose, but in any case, you can peel through and see what the different what each of the different compounds are made up of. What this is here is the linkage analysis. So each of these, so this is glucose again, but this time a dark glucose is a terminal glucose. A lighter one will give you 1,4, 1,3, 1,6, and so on. The same way here, looked at fructose or galactose, and so on. So the different shades of that same color make up then the different linkages. So in each of these contains one sample. Each of these columns will contain one sample, and in each of that sample we get all the linkages. And our method now will monitor over 100 different linkages. So we pretty much covered a lot of the different polysaccharides. And so when you have this kind of information, you can actually start grouping foods together. And so you can have a group 1 kind of food, a group 2, and each of these are based on their monosaccharide composition, right? So for example, a lot of the high starch foods will come out here. They're mainly glucose. You'll have foods here, for example, that are rich in different monosaccharides, rhamnose, arabinose, and so on. And you can sort of create almost different combinations. And this will become important later on, because what we eventually want to do is create diets that will then allow us to study the microbiome, have different diets, but at the same time maintain perhaps the monosaccharide composition. So now we go back to the potato, and as I told you before, each of these are really important. The blue here means these are glucose, and these are 1,4 linked. And so what you have then is you have the linkages of that. And then you'll have other polymers. So arabinogalactan is also in potato, and rhamnogalacturonan. But the other thing that you can get from this is that, look at this, this is the terminal glucose. This one has a lot more terminal glucose than this. And so, and this also has some branch glucose. So based on the kinds of linkages, this one, for example, will have more terminal glucose than this. And so you can start to infer higher order structures from just those composition. And I show that here. So this is rice. This is different kinds of rice. So there's a black rice, there's a red rice, different rice you can buy at the store. So the blue here tells you the amount of terminal glucose, and this is 1,4, right? So what that's telling you is that these guys here with the higher amount of terminal glucose have higher branching, whereas those with lower amounts of terminal glucose have lower branching. And the other thing that sort of comes out of this is this interesting rice. So all rice is made out of starch, it's 1,4, but then you have this black rice here that has a 1,3 link, which is really highly unusual. And you see that only in very, very small amounts here, but somehow here it seems to be amplified. Again, just showing you how, even though this is the same species, you can have very different types of polysaccharides. And so the idea here then, in the end, is that what we want to do is be able to take each of these different groups and map it to the microbiome that it corresponds to, right? And so, for example, these groups here would perhaps map out with certain microbes here. In the same way here, these groups here would map out with certain microbes there. But really what we want to do is sort of identify these symbiotic partnerships between all the different glycans. And we're doing this with David Mills' group, who's looking at a lot of the microbes in the microbiome work that we collaborate with. Okay, so, and this is some of the earlier results now. So for example, these is our plant fiber tree. And then what we show here, this is David Mills' results. What they showed, or what they found, was that if you take butternut squash, for example, create oligosaccharides from that, it turns out that you can actually selectively grow different bacterial strains with just butternut squash. So imagine then all the other kinds of polysaccharides that are going to be there. So this is, I'm sure, in the last few minutes, I'm going to show you the final problem. How do you figure out what are the polysaccharides in food? Because there's no single enzyme. It's not like proteomics where you have one enzyme that cleaves it into peptides. There's no one enzyme for all these different structures to give you all these different oligosaccharides. And so we came up with a method that we called FitDog, and that gives you, that degrades then all these different polysaccharides. And based on this now you can do an LCMS on, and then once you figure out what these structures are, you can then put it together and tell you what the parent structures are. This is just showing you different polysaccharides will have different signature. This is how the tandem mass spec tells you that we can tell what the structures are. And so then, for example, this is a coconut flesh. So coconut flesh, if you take our method with that, you get these peaks. And based on those peaks, we find that it has galactomannan, mannan, glucomannan, a lot of cellulose, and arabinoxylan. Yellow cornmeal has a lot of amylose. Yam leaves is amylose, arabinoxylan, and you can do the same thing and so on. And you can do this quantitatively. So this is different foods. And this is actually, the blue here is made up of cellulose. And then all the other polymers, the amylose and all these other polymers in there. But what it shows you is that a lot of our food is made up of cellulose, actually. So like, and also here, sort of interesting, this is that the broccoli florets versus the broccoli stems. And you can see that they're actually quite different. What's nice about this is that we also know what we don't know. So there are polymers in foods that we haven't figured out what they are yet, and there's no standards for them. And people haven't figured out what those structures are yet. So in any case, but in any case, you could see that we could start doing this. And so now we have this, so I'm gonna show you a project that we're currently on. And this is our NIH biomarker project with David and Bruce, Jennifer and Danielle, and then the graduate students in my group at least, who are involved in this. And so the question then is, how does a breastfed microbiota convert to one based on weaning foods? So we've already determined that each of these structures, and remember each of these, depending on the angle, will give you a different linkage. So each of these structures here will give you a very specific enzyme that will break this down. And so now if we go down to cell walls that make up a lot of our food and a lot of the carbohydrates, the question then is, how does that influence then the microbiome? And so one of the things that we want to know first is, what are the structures of food? So we went out and we set about looking at different weaning foods. So we looked at what mothers would feed their infants, and so we've characterized those. We've also looked at all the different commercial weaning foods that came out to about 500 different foods. And this is their monosaccharide compositions quantitatively with absolute abundance. And this shows you then all the different types of food that are in weaning, right? And so what we do with this then is we pick out, let's say, arabinose, which foods are rich in arabinose. We can do the same thing with fructose. And then what we do with that is we could align all the different amounts of arabinose that you may find. And then, so here's an example. This is an apple. We determine the water content. We know the dry mass. And then we figure out that it has based on 32 grams, we can get 32 milligrams of arabinose. This is raw beet versus steamed beet. We could tell you what happens when you cook it, for example. We can get the monosaccharide composition of that. And remember, we also can get linkages and even polysaccharide, but I'm just showing you the monosaccharide. And then this is raw pea versus steamed pea. Then you could take, say, an apple. You could take the skin, the flesh, and the hull. You can get those monosaccharide compositions separately, get the arabinose content, and then you can puree it and get the same kind of thing. So you could do this with all those different foods. And so now you could create a menu. And remember, these are kids, so we can't change their menu too much. However, we can offer them certain things. So in this case, if we want to do a high arabinose day, this is what it would look like. For example, in a low arabinose day, this is what it would look like. So we can do the same thing with fructose or whatever monosaccharide we want to do. And so we can quantitate then how much arabinose. So we give the mothers a choice. So this is what one menu would look like. We can also alter the menu But this just shows you all the different arabinose and arabinose content. So this is a second menu. So the mothers can mix and match. They have snacks here. We can create snacks for high arabinose day, or just a menu for another high arabinose day, and then a low arabinose day. And then what we're going to do is really look at the feces. We're going to look at what happens to the carbohydrate structures that are produced, and also a lot of other metabolites within the feces as we feed them. When they switch back and forth to, say, between arabinose and arabinose days. So we think this is an area that we're going to be doing. Well, we know this is what we're going to be doing. I'm just going to conclude with this, but I'm just showing you that there's now new and rapid throughput methods for the characterization of carbohydrates. The characterization of carbohydrates, we think it's going to be able to advance microbiome research, and that the structures of food are really necessary to do this. This is our group from maybe 2 years ago. I've been very fortunate to have a very enthusiastic and a very hardworking group, our funding agency. And I'd like to, with that, thank you all for your attention.
Thank you, Dr. Labria. For any questions, please drop those in the Q&A. I'll start with one. You know, as a mother of 3 who's weaned a couple infants, that's a lot of information to take in of all the different ways you're affecting the early gut microbiome. As you step back for a second, I mean, you know, in your study, I don't know how standardized the weaning age was, but I was curious in thinking about the development of the microbiome, how much you feel like, you know, weaning age, which does vary across various households and cultures more generally, how big a factor is that compared to, you know, the choice of foods upon weaning in terms of the ultimate kind of composition of the gut microbiome, if that's known, or what you might expect?
Yeah, that's a great question. I mean, one of the reasons why we chose the milk-fed infant is because we know how that starts, and for the most part, we know the kinds of we know the microbiome associated with that. We've looked at other weaning infants. We had a Gates trial, for example, looking at in Indonesia and looking at biomarkers for weaning foods, and what we find is that we didn't look at specific the bacteria, but we looked at the products, and the products that come out seem to adapt depending on when the weaning food, not necessarily depending on when the weaning food, but when the weaning food is actually introduced. So for example, we'll see something, we'll know when someone is feeding so fruits, for example, because initially we'll see that rise up. But then what we see is that then the fructose level actually goes down, and that didn't seem to matter what age they were in. So my suspicion is that it doesn't matter, the age perhaps doesn't matter as much as that you actually start weaning, at least for the first 6 to a year and a half or so.
All right, a couple of questions have dropped in the chat, so let me try and get to a couple before we switch speakers here. So from Byron Smith, does physical accessibility of polysaccharides interact with sugar and linkage composition when it comes to the microbiome degradation? Is there a quantitative way to measure those physical properties in foods as well?
Yeah, I mean, that's really important. And then it's the polysaccharide is actually less accessible than the, say, the oligosaccharides. And we've seen that when we start feeding it to bacteria. And so we think things like processing actually breaks down the polysaccharides to give you oligosaccharides. We're looking into that now, but certainly things like accessibility and is associated with processing, and I think that is one of the things that are going to be really important, is how accessible these are. The other question is, if you have a polysaccharide that's, say, combined with another polysaccharide that is not as digestible, how does that affect? Yeah, so there's a lot of these questions. I think that's really interesting, that should be further addressed. But I don't have yet a single parameter that says, you know, this one is more accessible than this one is. And I really think it depends on the enzymes that are associated with the microbiome and what's actually there.
Great. We'll do a final question, and then I'll let you answer some in the Q&A and one in the chat. But from Sophia Ramos, are there any specific bacterial groups which you think will feed on arabinose specifically?
Yeah, we've actually found, so we've done some proteomic analysis, and we found actually enzymes in the feces that are that we can go back to and figure out which bacteria they belong to. Some Bacteroides, for example. But I'm sorry, I'm not a microbiologist, so these names are sort of a little bit foreign to me. But we do have some ideas of what they are, and we can go back and figure out what they are.
Wonderful. Well, thanks again for joining us.
Yeah, my pleasure.
So it's as we're switching slides and such, it's my pleasure to introduce our next speaker, Dr. Martin Blaser, who's joining us from Rutgers University, where he's a professor of medicine, pathology, and lab medicine, and holds the Henry Rutgers Chair in Human Microbiome Research. Dr. Blaser is a very well-established and accomplished scientist with membership in both the National Academy of Medicine and the American Academy of Arts and Sciences, and he served as chair of the Presidential Advisory Council for Combating Antibiotic Resistance Bacteria, holds many US patents, and wrote the book Missing Microbes, which has been translated into 20+ languages. He's a physician and microbiologist, and his research has focused on understanding the relationships we have with our persistently colonizing bacteria. Work in his lab has been broad-reaching in scope and impact, and generally uses multi-omic approaches to understand microbiome changes that might fuel a variety of human diseases and conditions, including obesity, IBD, asthma, allergies, cancer, and others. And he'll be talking to us today on the immune consequences of perturbing the early life microbiome. So welcome, Dr. Blaser, and thanks for joining us.
Yeah, Tiffany, thank you very much for your kind introduction. I want to thank Peter and Susan for their invitation. And it's such a pleasure to participate in a symposium with so many talented and accomplished scientists. I'm quite humbled. I assume everyone can hear me and
yes,
you can see
great.
my slide? Okay, thank you. So today I'll talk about the immune consequences of perturbing the early life microbiome. And I think I'll be able to pick up on many of the themes that other speakers have talked about over the course of the day. So my talk's going to have 4 parts. Here's the first part, the theory. So I have been interested in something that all of you are aware of, and that is that diseases are not staying constant. There are diseases that are rising. For example, juvenile diabetes, which is doubling every 20 or 25 years, or asthma, which has gone through the roof, or reflux diseases of the esophagus, which have gone up dramatically. And there are maybe 10 other diseases that have gone up in parallel. So the question is, if you have 10 diseases and they've gone up at the same time, do they have 10 different causes, or could there be one root cause underlying all of them? And that's really my hypothesis, is that this involves the change in the microbiome. There are many things that have been changing the microbiome. We've heard about a number of them today. I'm going to focus on just one, which is antibiotics, because I think that has the scale to have a major effect. Worldwide, there are more than 73 billion antibiotic doses used a year in people. That's more than 10 antibiotic pills for every man, woman, and child in the world, and the numbers are going up. In the US, the CDC recently counted 258 million courses of antibiotics, 833 courses per 1,000 people. That's 5 courses for every 6 people, pretty constant. Importantly, in early life, children are getting almost 3 courses of antibiotics by the time they're 2, 10 courses by the time they're 9. Pregnant women, more than half of them are exposed to antibiotics during pregnancy, just before the critical intergenerational handoff of microbes that we've heard about from Ruth Lay and other speakers. And there's antibiotic exposures from farm use. We don't even know the magnitude of it. So many people have been concerned about the ecological effects of antibiotic exposures. And I consider it like the proverbial iceberg. The tip of the iceberg is antibiotic resistance. We've known about that for a long time. But I think that the body of the iceberg is its effects on disrupting the microbiome, which may have clinical consequences. These consequences could be transient or long-term. They could be developmental, situational, senescent, or generational. And they could involve immunologic properties, metabolism, neoplasia, maternal effects. Today, I'm going to focus just on one of them, which is their immunological effects. So part 2, evidence of correlation of early life antibiotic exposure and subsequent disease risk. Recently, working with colleagues at the Mayo Clinic, we have conducted a study to assess the associations of early life antibiotic exposure with diseases that manifest in childhood. This was recently published Versa et al. And so in this study, we looked at a period of exposure during pregnancy and during the first 2 years of life, and then a period of health outcomes from the age of 2 to the age of 13. Here we studied all the children born in Olmsted County, Minnesota, which is where the Mayo Clinic is, over about a 9-year period. And we had to exclude some. We ended up with about 14,000 children, more than 100,000 person years to be analyzed. Here are the characteristics of the studied children relative to antibiotic exposure in the first 2 years of life. In total, 14,000 kids, about 10,000 got antibiotics in the first 2 years, about 4,000 did not. The number of courses of antibiotics ranged, in those who got them, ranged from 1 up to more than 5. There were 3 dominant types of antibiotics that were studied. And I want to point out that there were about equal numbers of boys and girls. So there, in essence, are 2 parallel studies. So here we'll look at the Kaplan-Meier analysis of time to developing asthma by sex and by antibiotic exposure. Here we're looking at children from the age of 2 after the antibiotic exposure period. This is the health outcome period up to the age of 14. Here's the probability of asthma. The red lines are girls. Here are the girls who did not have antibiotics in the first 2 years of life. Here are the girls who did. Here are the boys without antibiotics. Here are the boys who did have antibiotics. You can see exposure to antibiotics was associated with a much higher rate of developing asthma. In fact, we looked at 10 common health conditions in childhood, such as asthma, allergic rhinitis, food allergy, atopic dermatitis, et cetera, including metabolic, immunological, metabolic problems, and also neurological problems. And here's the adjusted hazard ratio in these studies, in which the hazard ratio of 1 is neutral, there's no association. And of these 10 problems, all of them are, the hazard ratio is greater than 1, and 8 out of the 10 are statistically significant, and a couple of them just miss. So just to summarize this study, considering what we found is that antibiotic exposure, number of courses, timing of exposure, and specific antibiotic classes were all associated with subsequent risk for 8 important childhood diseases. But of course, this is correlation. So part 3, causal roles for antibiotic-induced perturbations. Well, we've been doing a number of studies over the years, and I want to start with this study done by Victoria Ruiz when she was a postdoc in the lab. Victoria gave mice either 3 courses of antibiotics to mimic 3 courses of antibiotics in a child, or 1 course, starting very early in life, mouse day of life 5. And here in this principal coordinates analysis, we're looking at the microbiome on day 52. Now, this is more than 40 days after that single course of antibiotics. Here are the controls. Here are the mice that received antibiotics 1 or 3 courses. You can see the microbiome is still quite perturbed as a result of that early life exposure. Now, there were many parts of her study. I just want to focus on the effect of the antibiotic on allele gene expression and immunological effectors. Here's a heat map of about 500 different inflammatory and immune genes in the ileum. This is based on gene expression. Here's an unsupervised hierarchical clustering. Black are the mice that didn't receive antibiotics. Pink and green are the ones that did. You can see that they cluster together. There are many genes that are differential here. In total, 147 different genes. This is, by the way, about 50 days after the antibiotic had stopped, after the single antibiotic had stopped. It included important genes like the polymeric Ig receptor. There were effects on Th17 cells. There were effects in intestinal secretory IgA as well from the antibiotics. And what I don't have time to show you, but you'll have to see the paper or take my word for it, that the antibiotic perturbed microbiota was both necessary and sufficient for the altered host responses. So this is markers of immunity. What about illnesses or diseases? Well, here's an epidemiologic study looking at the likelihood of IBD in Danish children by early life antibiotic exposure. This is the reference group, no antibiotics. The more courses of the antibiotics they got in the first year of life, the more likely they were to develop IBD. So we wanted to know, can an antibiotic altered microbiota affect IBD outcome? So we studied this in wild type mice in the model of DSS colitis, as was reported earlier today. This is work that Saren Oskol did. So she exposed mice to the macrolide tylosin or not. And then later, here about 2 weeks later, they were exposed to DSS or not. And we looked at a number of different phenotypes. Here we're looking at body weight. The mice that did not get DSS, they continued to gain weight. The mice that got DSS water and then DSS, they lost weight. But if they had antibiotics and then DSS, they lost even more weight. So this accentuated the colitis. We looked at the tissues, and if you look here at the aggregate scores, the most inflammation was in the mice who got DSS, but it was more if they had been exposed to antibiotics earlier than not. And we see it in each of the individual scores. So was this an antibiotic effect, or was this through the microbiome? So to answer that question, we did a transfer experiment. So from mice that received antibiotic or not, 30 days after the antibiotic was over, we sacrificed the mice. We obtained their cecal contents and we gavaged them into germ-free mice. And then we exposed these mice to DSS. And here we're looking at the body weight change. These are the mice that received the normal microbiota. There's their weight change. And here are the mice that received the perturbed microbiota. So this provided evidence that the antibiotic perturbed microbiota was changing the phenotype of colitis. Now we took it another step further, and this is where we wanted to know what can an antibiotic-altered microbiota affect IBD outcome in the next generation. So we did these studies in IL-10 knockout mice. These are mice that spontaneously develop colitis. This work was done by Angel Schulfer when she was a graduate student in the lab. Angel gave antibiotic-perturbed microbiota or normal microbiota to germ-free mice that were pregnant. And now she conventionalized these mice. They were either wild type or IL-10 deficient. They gave birth to their pups, and then we studied the pups when they were middle-aged. We were interested in the effects on the ecology and the effects on disease. There were many effects on ecology. You can read those in the paper. I want to just mention the effects on disease. So here we're going to look at the colonic pathology in IL-10 knockout pups at week 21, according to the microbiota to which their mothers were exposed. This is the colon from a mouse whose mother received the control microbiota. There's inflammation here, which we would expect in a middle-aged mouse, IL-10 deficient mouse. But here's the colon from the mouse whose mother received the perturbed microbiota. It's much worse, and in fact, it's about 30-fold worse statistically. This difference is quite representative. To summarize the key points of this experiment, I want to remind everyone that neither the pups nor their mother received antibiotics. So therefore, the enhanced disease signal is entirely microbial. That means that antibiotic effects cross generations. And it also means that inheritance also is based on microbes and their genes. Now, we wanted to study the mechanisms a little further. And here we're looking at assessing the effects of the early life antibiotic exposure on colonic immune phenotypes. This is work done by Xiaoxiao Zhang, Tim Borbet, and Angela Fellenger. It's a partnership that we've had with Ann Mueller's lab at the University of Zurich. We did a number of different experiments, but today I'm just going to talk about when we gave newborn mice ampicillin, the most commonly used antibiotic in childhood, or not. And then we sacrificed them. We looked at their microbiome and we immune phenotyped them. From these, we also did a microbiota transfer to germ-free mice. And here we're going to be looking at immunological phenotypes. So first, we look at the microbiome. This is in the mice that received antibiotics or not. When we look at the beta diversity, we see the microbiome is quite different, even 35 days after the antibiotic is over. The alpha diversity is a big decrease, all as we expected. Here's an unsupervised hierarchical clustering, which is clearly dividing the antibiotic perturbed microbiota with the normal microbiota. And there are many organisms that have disappeared here with the antibiotics and that have not come back 35 days after the antibiotic is over. So the effects, like in Victoria's studies, are long-lasting. Now we're going to look at colonic lamina-propria T cell populations in the antibiotic exposed or at the control mice. And we're looking at them at several different time points. And what here, as we're focusing on CD4 T cells, which are down in the antibiotic group, T regulatory cells down, and other CD4 effector cells also down. What's important to note is that we did not see any defects in any of the colonic myeloid cell lines or in T cell populations elsewhere in the lung, the stomach, and the mesenteric lymph nodes. This was the target of the perturbed microbiota. We looked further because we were interested in T regulatory cells. These might either be neuropilin positive. These are the thymic derived T regulatory cells. There was no significant difference. But in the neuropilin negative Tregs, which are the peripheral Tregs, there's a strong effect, and especially the subset that's also expressing ROR gamma. So the question is, why did the mice have this big defect? And so one idea is that the antibiotic altered microbiome was signaling differentially to the epithelium, especially to the dendritic cells, especially to tolerogenic dendritic cells, which instruct CD4 cells, which recruit CD4 cells and instruct them to become peripheral Treg cells. So we studied this question by asking whether dendritic cells from antibiotic treated or control mice would induce FOXP3 expression in co-cultured CD4 cells. And the main result is shown here. The dendritic cells that came from the control mice strongly converted the CD4 cells into Tregs, and this was much less in the cells from the ampicillin treated mice. And so this is consistent with the idea that the antibiotic-induced dysbiosis is compromising the tolerogenic activities of the DCs, leading to the P-Treg deficiency. So then we wanted to know, again, is this just a direct antibiotic effect or is it a microbiome effect? So we did a transfer experiment that asked really the parallel question. And here the donors were mice 3 weeks after the antibiotic or not. The recipients were germ-free mice. And we're now looking at phenotypes 4 weeks after the effect. Again, the microbiota that was transferred is different. And again, we see these decreases now in actually thymic Tregs, peripheral Tregs, and the subset with ROR gamma T. So the question is, now that we've shown these differences in the colonic P-Treg cells, what are the functional consequences of this deficiency? So we then began studies looking at the effects of the early life antibiotic exposure on allergic manifestations in an antigen challenge mice. This again was in partnership with the Ann Mueller lab. This is work that Tim Borbet did when he was a graduate student in my lab. Tim started with mice from birth and from days 5 to 9 gave them either azithromycin or amoxicillin, the two most commonly used antibiotics in childhood in the United States, or not. And then at day 46, he sensitized and challenged the mice with house dust mite antigen. This is an important antigen in allergy models, or as a control with PBS. And there were intermediate sacs so that we could study intermediate phenomena. We looked at the microbiome and we looked at immune profiling. Here we're looking at the effects of the early life antibiotic exposure on pulmonary markers of allergy. Here we're looking at the total number of cells in bronchoalveolar lavage, the proportion that are eosinophils, the proportion that are neutrophils. The PBS are the mice that were in the control group. They didn't get the challenge with the house dust mite antigen, and all of them have very low numbers of cells. But the mice that got the house dust mite antigen had higher numbers of cells as expected, but it was higher in the group that was pre-exposed to azithromycin than control. Total cells, eosinophils, but for neutrophils, it was the group that received amoxicillin. When we looked at some markers of genes involved in IgE and eosinophils, we saw significant increases here in genes involved in recruitment and chemotaxis of those cells and phenotypes. Here we're looking at IgE levels in relation to challenge and antibiotic exposure. In the serum, higher IgE in the mice that received azithromycin. Also in the lung, they correlated very well. Here we did in vitro, ex vivo work, stimulating CD4 cells. No real change in the total population of CD4 cells, but the proportion that are IL-13 positive increased with azithromycin, and IL-17 positive increased in the proportion the mice that received amoxicillin, consistent with the neutrophilic phenotype we saw before. So we wanted to see, could we transfer this again to germ-free mice? And when we did an experiment of transferring the microbiota, the perturbed microbiota or the control microbiota to adult germ-free mice, we found no phenotype at all. And we concluded that it was because those mice were too old. The window of opportunity had passed. So from another group of germ-free mice that had been that we've had the microbiota transferred from, we bred them and then we studied their pups. So their pups were exposed to the altered microbiota from birth or not. And we did the same kind of sensitization and challenge. And here are the results for several important markers. And we see that there's increased serum IgE, even in the PBS group with azithromycin, with the HDM, total cells in the BAL up compared to controls, total eosinophils compared to controls. Here we didn't see a neutrophilic phenotype. So again, the antibiotic is working by perturbing the microbiota, which is altering all these changes in the lungs. Finally, in this experiment, we wanted to see, could we give these mice asthma? So we did methacholine challenge trials to assess airway resistance and elastance in the PBS group or the HDM group. The blue line is the control group. These colored lines are the antibiotic groups. The mice that were exposed to antibiotics had more airway resistance in both, whether they were unchallenged or challenged. So this affected the tone of their airways, and there was some increase in elastance in the PBS group as well. So this work showed to us that, well, let me just summarize and say that from all of this work, we could summarize and say that antibiotics have long-term effects on inflammatory and immune cells. The effects are due to perturbing the microbiome. Other factors of modern life also contribute, but I haven't discussed them at all. Effects may be transmitted to the next generation, and we need to find and implement solutions. So the last story I want to tell you about is about some hope. So this concerns models of type 1 diabetes. Shusang Zhang looked at the effect of these pulsed antibiotic courses, therapeutic courses on type 1 diabetes development in NOD mice. He gave mice 3 courses of antibiotics or control, or even a single course of antibiotics, and then monitored diabetes outcome. Here's the Kaplan-Meier analysis. The blue lines are the control mice. They are developing diabetes as expected, but the mice that got antibiotics are getting even more diabetes, and it's happening faster. And this confirmed an earlier study as well. Now, Shusong showed many things which are in his paper, but I just want to focus on one change, which is the effects of gene expression in the ileal wall, looking at the toll-like receptor signaling pathway. Here's the pathway. Every gene that's in color has been changed by the antibiotic exposure. So there's the signal is being transduced into the ileal wall, similar to Victoria's changes that I showed earlier. So what are we going to do? We're not going to get rid of antibiotics, but maybe we can use them more wisely. But the other possibility is that we have to develop restorative strategies after antibiotics are used. So Shusong has more recently done another experiment where he wants to see, can he restore health or restore the baseline using a microbiota transfer of cecal microbiota. So in addition to the first group of the control mice and the mice that received the antibiotics, he had a third group. In the mice that received antibiotics, he gave them cecal contents from moms on the day of birth, which we call cecal microbiota transfer, or CMT. So here's the Kaplan-Meier plot. The blue line is the control group. The red line is the group that received antibiotics. They have more diabetes as expected, but the group that received antibiotics and then got the transfer, they're just about back to baseline. So we have a model in which we've been able to restore the phenotype to baseline. And that's useful so we can discover important properties. When we look at the microbiome, we see that the CMT group is intermediate between the control and the antibiotic group in microbiome richness. Evenness is actually at the control level. In the beta diversity, here's the antibiotic group, here's the control group. The CMT group is intermediate, so there's partial restoration, but it's enough to restore the phenotype. We're able to identify a number of important organisms that some others have mentioned before and that we're quite interested in studying. We've looked at the metagenome. And this slide shows some of the metagenomic effects of 1-PAT and restoration. Here we're looking at the global metagenome. Blue is control, red is PAT, green is the restored group. So we can see that there's an intermediate global position. When we do a heat map, with an unsupervised clustering, we see that here is the antibiotic, essentially the antibiotic group that got the PBS as a control, and this second cluster are the control mice or the mice that got antibiotics but received the mom treatment. And we can identify a series of different pathways in which the antibiotic effect in the metagenome is lost, and it is the control effect is seen. And from these pathways, we've done a number of metabolic analyses, which I don't have time to show you. But we have looked at gene expression in the ileum. And here, this is the difference.
Dr. Blazer, thanks so much. We're just getting a little short on time, but for a couple minutes left. Thank you.
Okay, thank you. So unsupervised clustering, bottom line is that the CMT group is clustering with control, not with PAT. And so we can identify these genes. We have a couple of very important genes that we have found. We have looked at the epigenetics, looking at histone H3 modification. This is involved in chromatin accessibility, and this lysine 27AC increases activation. It's up with antibiotics, but it's restored after the transfer. And this alternative one is down with antibiotics and up again. So there's an epigenetic effect. And now we're looking at microRNA. And basically we see again that the CMT group is clustering with control and not with PAT. We know many of the mRNAs and they're linked to some of the important genes. In summary, an overview of this restoration study is that this provides a pathway for discovery of microbes, genes, metabolites, host genes that are perturbed by antibiotics that drive autoimmunity and that can be restored. I'm sorry to run so long. Thank you.
That was awesome. Thanks so much. I have a bunch of questions I can ask you, but I'll move to those in the Q&A given time. The first is, can you comment on the potential for non-microbial aspects of any donor material, such as nutrients or metabolites or cells, to affect the transplant recipient's immune response?
Yeah, I mean, that's the problem with any kind of fecal or cecal transfer, is that it's a very biologically complex substance. But this allows us pathways for discovery, and if we find phenotypes, then we can try to. And so that's why we're very interested in the specific microbes that are differentially lost and then regained, considering that they may be the great targets.
Another question was, have there been any attempts to rescue immune phenotypes with FMT or probiotics clinically? And if so, do you think this would be an important standard of care for antibiotic treatments?
I mean, as you know, if you go to clinicaltrials.gov, there are like more than 300 trials on FMT. This was before COVID. I don't know what's happened since then, but people are trying just about everything. And the idea of some kind of restoration after antibiotics is a highly prevalent thought. The question is, what's the right thing to do? And my hope is that from doing these mouse models, we can discover the right things.
Thank you. There's one more in the chat if you want to answer that one, but thanks again for joining us. That was really a wonderful talk, and I'm going to turn it over to Sue Lynch here to introduce our final speaker.
Thanks so much.
Great, thank you, Tiffany. Thanks, Marty. It is my absolute pleasure to introduce our final speaker of the day, Dr. Ami Vat, who is an assistant professor of medicine and genetics at Stanford University. She's also the director of the Global Oncology, a nonprofit organization based in the Center for Innovation and Global Health. And Ami's group really seeks to improve outcomes in patients with hematological malignancies by characterizing the dynamics of the microbiome in immunocompromised individuals and exploring how changes in the microbiome are associated with idiopathic diseases in this population. And today, her talk will be entitled, Measuring What Matters in the Microbiome, A Path to Precision Medicine. Ami, it's all yours.
All right, well, thank you so much. And thanks to all the previous speakers who really gave fantastic talks on a wide range of topics. I'm going to try to weave in some of the themes that we've heard about over the course of the day. Just to get things started, these are my disclosures. And the most important slide of my presentation, which is a picture of my group. I'm really fortunate to work with a wide variety of extremely talented and engaged scientists, and I will be presenting many of their projects over the course of the coming about 25 minutes. I'll try to call them out as I present their work. So this is a beautiful photomicrograph of a cross-section of a colon of a mouse. This was taken by Kristen Earle when she was a student in Justin Sonnenberg's lab at Stanford. And what you can see here is on the top left, a bunch of microbes, on the bottom right, the DAPI counterstained nuclei of the colon, and in between the green stuff are 2 layers of mucus, thin and thick. We know that the microbiome is incredibly important for things that range from barrier function to immune modulation, to infection, to nutrient extraction. And for the most part, many of the efforts that have been carried out to date have focused on asking the question, who's there in the microbiome? Over the course of today's fantastic presentations, you've heard from other investigators about how microbes can actually be transmitted from mother to child. We've talked about how that can actually persist over generations and even millennia. And we've learned about how microbial communication between mother and newborn or mother and neonate can actually be critical in shaping the newborn. We've learned that dietary choices can dramatically impact the microbiome, not just human milk oligosaccharides, but also plant polysaccharides and plant-associated microbes. And we've heard about how the microbiome, antibiotics, malnutrition, and immune maturation are all intricately linked. So what do these different things have in common? The transmission of microbes vertically, the impact of diet on the microbiome, the relationship between antibiotics, early childhood malnutrition, and outcomes? Well, at the core of all of these different scientific studies, and I would argue any scientific study, are common measurement tools. And so what our lab seeks to do is to ask the question, can we make better measurement tools? Now in microbiology, we have a long history of measuring microbes. For hundreds of years, we've been able to isolate and culture microbes in petri dishes, thanks to the work of people like Robert Koch. We can then stain them and look at them under the microscope, thanks to the work of Graham and Leeuwenhoek. And most recently, over the course of the last 20 or so years, we've been able to sequence microbes. This sequencing revolution has enabled us to first learn a lot about microorganisms that we can't culture through the enumeration of ASVs or OTUs, operational taxonomic units. And over the course of the last couple of years, we've seen an explosion in what is possible with shotgun genomics, leading us to the era of MAGs, or metagenome assembled genomes. Fundamentally, we believe that precisely measuring microbes will enable precision medicine. And so I'm going to tell you about 4 different vignettes that'll kind of highlight different aspects of tools that we've developed and how we've applied them to try and answer some interesting questions. So first, measuring microbes. In this vignette, I'll talk to you about how we think the gut microbiome is a major source of infections in patients who are undergoing hematopoietic cell transplantation. As Sue mentioned in the introduction, a major focus of our lab is trying to understand what causes mortality and morbidity in patients who are undergoing hematopoietic cell transplantation, also more commonly known as bone marrow transplant. Now, many of you may know that bone marrow transplant is a fantastic treatment for people who have refractory or relapsed hematologic disorders, but unfortunately it is a bit of a deal with the devil. And about 20 to 30% of our patients unfortunately die in the first year after transplantation. Now, a subset of these patients will die due to relapse of their original disease or graft versus host disease, which is an immunologic sort of disease. But a good proportion of people actually contract infections and die of infections. This is exemplified in this case. This is a patient we actually saw at Stanford University Hospital. A 39-year-old woman came in with B-cell acute lymphoblastic leukemia. She underwent a hematopoietic cell transplantation, and 37 days after her transplantation, she developed fevers and chills. Now, you don't need to be a doctor to presume that she might have an infection, and indeed, a blood culture grew out Staphylococcus epidermidis. The question we and others had was, where did this infection come from? Now, if you followed the work of Tiffany Scharschmidt and others, you will know that Staphylococcus epidermidis is a common skin commensal, but we decided to dig a little bit deeper. This is the work of Tessa Anderman, a former postdoc in my lab who now has her own group at UNC Chapel Hill, and Fiona Tamburini, a former graduate student in the lab who's now at Genentech. The simple question that they wanted to answer was, are these bloodstream infections that our patients are getting coming from the hospital environment, or might there be an internal source? Understanding that the gut microbiome is our largest and most dense exposure to microbes. To answer that question, Fiona wrote a really nice and straightforward bioinformatic tool called Strain Sifter. What it does is it identifies highly concordant microbial strains between samples. In this particular case, we compare stool samples collected prior to infection with the actual bloodstream bacterial isolate that was cultured out of patients' blood. What this led us to investigate was whether or not the gut might be a reservoir of bloodstream infection pathogens in patients undergoing hematopoietic cell transplant. So in the patient case that I shared with you earlier, this is actually a stack bar histogram of the gut microbiome composition of this individual 1 day prior to her bloodstream infection. As you can see, different colors mean different organisms, and you'll see a very thin little layer, which is Staphylococcus epidermidis. But you would be right to ask whether or not that's really the same strain. We know that there are multiple strains of Staph epi on people's skin, for example. So just because we see a Staph epi in the gut and one in the bloodstream doesn't mean that they are identical. We applied Strain Sifter, and one of the outputs of Strain Sifter is a phylogenetic tree. Just a quick orientation to this, the closer things are on that tree, if they're on the same branch, they're basically indistinguishable from one another. I've highlighted the patient 13 samples in blue here, that's this particular patient, And what you'll see is that the bloodstream isolate and her stool sample 1 day prior to her bloodstream infection are actually indistinguishable from one another, suggesting that staph epidermidis in this particular case that made it into her bloodstream may have originated in the gut microbiome and not in the skin. Now, in this particular case, we did not have skin swabs, so we don't actually know whether or not her skin harbored this strain, but certainly we know that her gut microbiome did. Now, this was not the only example of this sort of thing that we found. We also found that diverse bacterial infections can originate from the gut or be present in the gut prior to infection. This includes Enterococcus faecium, E. coli, Klebsiella pneumonia, and Strep mitis, all of which are known to be inhabitants of the gut microbiome, although I will note that Strep mitis is typically thought of as more of an oral microbe. By contrast, we didn't find this for everything. Staph aureus, for example, we were never able to identify in this particular study a Staph aureus bloodstream isolate that had a concordant strain in the gut microbiome prior to infection. Taken together, we found that approximately 40% of the bloodstream infections in our transplant patients were traceable to the gut microbiome. And beautiful onward work that was done by Tobias Hull at Memorial Sloan Kettering showed actually this same sort of pattern for fungal pathogens. Building off of this work, Ben Saranoshian, who's a graduate student in my lab, has been asking whether or not pathogens are actually transmitted to and from gut microbiome reservoirs. If it turns out that our patients are walking into the hospital with their pathogens in their gut microbiomes, well, maybe there's not a whole lot that we can do to try and prevent them from catching them. Alternatively, what if while they are in the hospital receiving things like antibiotics that are clearing out their gut microbiome, what if that actually sets them up to acquire pathogenic gut microbes from their environment or other individuals? This work is actually in progress. I'll give you a little sneak peek to say that we have found evidence of potential gut microbe transmission, both pathogens and commensals, but we actually find that transmission is much less common than we thought it would be. When I think about this holistically from a clinical translational perspective, the big question that we have is, can we use this information to manage patient microbiomes with the hope of preventing either the acquisition or the outgrowth of pathogens? Of course, much of the work that we do, including the vignette that I just shared with you, is focused on who's there. And I hope in this simple example, I've convinced you that strains matter, and being able to track different strains and differentiate them from one another is critical for being able to dissect the mechanisms by which disease occurs. It will not surprise this audience to know that most microbes have yet to be isolated and cultured, and this is not just microbes that live in extreme environments, this is also microbes that live in and on us. Despite this fact, fecal microbiota transplantation therapy is widely used for the treatment of relapsed refractory C. diff, and as Marty just alluded to, it's being explored for the treatment of a wide variety of other diseases. But this really is a black box therapy. When I take a look at a sample like this, these are fecal microbiota transplant capsules or so-called crapsules, we really don't know what's in them. Now, it would be great if we could, in a very painstaking way, isolate and culture every single one of these microbes, but we know that that would take a long time and probably wouldn't be complete. What if we could instead find a way to sequence all of these without having to isolate and culture and generate what's on the left side of this image, which are kind of nice completed Circos plots? That was actually our dream when I started my lab about 6 or so years ago. We wanted to get an orderly set of genomes as indicated by those outside colored rings in those Circos plots, just by sequencing these samples and applying some cool computational biology. We can kind of summarize the goal of this as saying we want to be able to perform culture-free microbial discovery and tracking. You all have heard a little bit about MAGs today, the idea of stitching together metagenomes to form genomes, taking short sequencing reads, stitching them together, growing them out into longer contigs, and eventually trying to get more completed genomes. We call these more completed genomes MAGs, which is short for metagenome assembled genomes. And many of you will know that over the course of a few months, actually, between 2018 and 2019, 3 landmark papers were published on this topic. In these 3 papers, they were able to go from having only about 160,000 bacterial genomes in RefSeq, our reference database, to adding 200,000 new metagenome assembled genomes from these 3 studies alone, which is pretty phenomenal. MAGs have really advanced our ability to try and understand the different strains that can be present in microbiomes. But of course, while they are certainly an advance, there are some problems with MAGs. For example, in this MAG that our lab generated from a healthy adult stool sample, you can see that the genome is highly fragmented. You'll also notice that there are 2 different shades of colors there. There's a dark green and a light green. It turns out that in the actual MAG that we built, all of the light green stuff was missing, which means that metagenome assembled genomes are highly fragmented and incomplete. And what is missing is often mobile genetic elements. Mobile genetic elements are fascinating, not for the least that they provide things like antibiotic resistance. And so not having mobile genetic elements incorporated into the genome is actually kind of problematic. To overcome this challenge, we thought long and hard about how we might develop methods to demystify these black box therapies, to enable strain tracking, and to generate better genomes from these metagenomes. Our first solution to this was developed by Eli Moss, a former graduate student in the lab, who's now at Hexagon Biosciences, and Alex Bashara, a former student in the computer science department at Stanford and a postdoc in my group who has since finished. What they did was they applied read cloud sequencing, also known as linked read sequencing, and a new assembler called the Athena assembler that Alex developed. What this generated was what is in this outside circle of this Circos plot. You can see in golden color, we were able to assemble the genome of this Phascolarctobacterium species in just 2 pieces. So far improving the contiguity and completeness of the genome compared to that metagenome assembled genome that I showed you earlier. Using these types of methods, Alex and Eli and a current postdoc in the lab, Sumaya Zlitny, asked the question, well, is it possible that there is evolution of microbes in clinical timescales, days and weeks, in our hematopoietic cell transplantation patient guts? These individuals are being exposed to a huge amount of different selective pressures, so it would be reasonable to think that things like antibiotic resistance might be developing de novo. We have longitudinal samples from hundreds of patients over about 5 to 6 years of time, and Sumaya and Alex and Eli decided to dig deep into just one. We've heard about how microbial evolution can actually take place over the time course of days and weeks and months and even lifetimes from Nandita earlier today. And I'm going to build on that and tell you about a simple case. This is our mystery case number 2, also a patient we saw at Stanford. So this is a 46-year-old man who had myelodysplastic syndrome, which is a pre-leukemic syndrome, also abbreviated MDS. He underwent a transplant at the time point that is indicated with the red line, and he became oligo-dominated, as you can tell, based on this stack bar histogram, by this blue organism. We performed read cloud sequencing, that approach that I described to get more contiguous genomes on this sample. And in the next slide, I'm going to show you the Circos plots for that blue organism over those 4 time points. Now, this is a very busy slide. I'm just going to call your attention to a couple of things. Each pair of rings represents that organism, which turns out to be a Bacteroides cacchi, at a different time point. So time points 1, 2, 3, and 4 in this particular case. In the blue is the read cloud assembly, which you can see still has a lot of fragments in it, but it is much more contiguous and complete than the black or gray rings just inside of each blue ring, which is the metagenome assembled genome from the same sample. If we zoom into particular locations within this genome, what we noticed was that oftentimes there were actually structural variations at these places where we got a good assembly from the Athena approach, but we did not get a good assembly from the metagenome assembled genome. And we're going to zoom in on just one of these particular examples. So this is the genomic location that we're going to take a look at. What you can see here is we have a bunch of genes that are indicated in red. I've highlighted one of the genes in a green box. This is NORM, a multi-drug efflux pump gene. And just to the left of it, you can see this other gene that I've labeled with the pink box, IS. IS stands for insertion sequence. This is also known as a transposon or jumping gene. This particular organism has tens of copies of this jumping gene that are variably present throughout the genome. Now, you'll notice that I've also put an arrow on that jumping gene, and the arrow points outward. This is an outwardly directed promoter, which should be able to drive transcription of the gene that is to the right of it, in this case, NORM. If we take a look at those improved assemblies that we get from this read cloud assembly, and we zoom in on this locus, you can see that at this time point, the first time point on the left, this IS sequence is not present. At the next two time points, the IS sequence is present. And remember, when the IS sequence pops in, you should have an outwardly directed promoter present. We then did RNA-seq, and as you would predict, once that IS is present in that location, this promoter seems to be sufficient to drive transcription of NORM. And concomitant with this, we were able to find that those strains that overexpress NORM, secondary to the insertion sequence being just upstream of them, were 2 to 8-fold more resistant to fluoroquinolones, suggesting that antibiotic resistance might actually be developing in clinical time courses of days to weeks through not SNP acquisition in this particular case, but actually movement of mobile genetic elements. So this is an example of what you can learn with more complete genomes and better strain level resolution. But of course, every silver lining has a cloud. And in this particular case, we had some bad news, which was that read clouds did well with some organisms, but they assembled some things really poorly. And I'm just going to give you an example of a result that really bummed us out. So I'm just going to draw your attention to this organism, Prevotella copri. We've heard about it earlier today. Prevotella copri is a very prevalent organism in the gut microbiome. It's a relative of Bacteroides species. And you can see that in this particular sample, we've got N50 on the x-axis. N50 is a measure of genome contiguity. If you have a perfectly assembled genome, the N50 should be the size of the genome. And you'll see that we do very poorly. The full genome should be about 3 or so megabases. To overcome this challenge, Eli Moss, who I introduced earlier, and Dylan McGinney, who's a current graduate student in the lab, decided to apply long-read sequencing or nanopore sequencing and a new assembly workflow that Eli developed called LATHE to try and generate more complete genomes with this approach. I won't belabor the details of this. It took us a long time to be able to figure out how to extract enough high molecular weight DNA to be able to do this, but we were eventually able to do the nanopore sequencing. For those who aren't familiar with nanopore sequencing, it works very differently than standard short-read sequencing. Standard short-read sequencing is actually chemically very similar to Sanger sequencing. It just happens on a solid phase. Whereas nanopore sequencing pulls a single strand of DNA through this pore. Electrons flow from the top of the membrane to the bottom of the membrane. And in this narrowest part of the pore right here, electron flow is impeded by whatever sequence is present. What is measured by nanopore sequencing is then electron flow, which is present as this squiggle graph. And that squiggle graph is turned into a sequence by a machine learning algorithm. So using this approach, we were able to generate the first complete and closed P. copri genome from a human. Mick Watson's group actually very close to the same time generated the first complete closed P. copri genome from a cow. This is what it looks like. It has nearly 100 transposons in it of about 5 different flavors, which explains why it was so hard for us to assemble with other methods, suggesting that transposons may actually be a very important part of how Prevotella adapts, and is so successful. We've taken these methods forward. We're applying them to a variety of other types of samples. One thing that I won't have time to talk about, but I put in some references here, is a really exciting collaboration that we have with the H3 Africa Genomics Consortium, where we're trying to better understand the composition of the microbiome of individuals living in rural and urban Africa. So we've talked about who's there, and I hope I've convinced you that strains matter. They matter not only for human health and for infection, but as you heard earlier from Jeff Gordon, we're really interested in trying to narrow down, for example, which bifidobacterium strain did they exactly find that ended up improving malnutrition outcomes in individuals. So this is an example of how we can use metagenomics to try and improve the resolution of our identification of medically useful strains. But of course, beyond who's there, there's a fundamental question of what these organisms make and how what these organisms make may be related to these clinically important things ranging from barrier function to nutrient extraction. What they make actually can communicate across this host-microbe interface. And we know that microbes use small molecules and proteins to communicate within species, between species, and even between domains of life. Now, nature has provided us with a huge amount of chemistry to fill our medicine cabinets. Compounds such as doxorubicin and bacitracin have been derived from bacteria. I would argue that small molecules have been relatively well mined for their purposes in medicine. And we heard from Mohammed earlier today about how bacteria can actually modify small molecules in a variety of exciting ways. But proteins are optimal signaling biomolecules in some way. They're diverse, they're highly specific, they're biocompatible, and you can synthesize them scalably, but they're undermined despite the fact that I think that they're optimal biosignaling molecules. Why are they undermined? Well, they're usually left out of bioinformatic annotation algorithms. And they're hard to detect experimentally. They run off gels and columns, and when you do things like metaproteomics, the likelihood of detection is proportional to the size, meaning that we usually miss them. Gila Sparrow, a former postdoc in the lab, sought to overcome this challenge and recently annotated as comprehensively as we could small genes in these metagenomes. This work was published about 1.5 years ago, and I'll summarize it here. We identified 4,500 new families, most of unknown function, 96% of which have no known protein folding domain, 48% have no homolog in RefSeq, and about 30% are involved in cell-cell communication. Brian Freeman, a graduate student who just defended his thesis about a week ago, developed a way to validate these predictions using MetaRiboseq, which validates well against gold standard RiboSeq and has signal that's enriched within the codons. Just to summarize, he was able to identify that over 1,000 of these predicted small open reading frames were truly transcribed and translated with just a single set of experiments on 4 fecal samples, suggesting that these predictions are correct. I'll just end by saying, if you study bacteria of interest and you want to understand what small proteins might be encoded, you might want to check out a paper that was just published by Matt Durant in my group. What he did was write an annotation algorithm that enables us to automatically annotate small proteins using a profile HMM and deep learning based approach. The tool is called SmortFinder and it's available online. So I'll close by saying that we're interested in how small microbial proteins may modulate things ranging from immunotherapy efficacy to quorum sensing in pathogenic bacteria. And we hope that we can achieve this objective of precisely measuring microbes so we can enable precision medicine. I talked about how the gut microbiome is a major source of infections, how we can cure and treat diseases using black box microbial therapies. Using these 2 different genome assembly approaches, we can now demystify these black box microbial therapies, and I ended by talking about small proteins. So with that, I'll just say that our goal is to now connect these things. And with that, I'll thank my lab, our collaborators, our funding sources, our recent grads, and most importantly, the patients and study subjects who participate in our work. Thank you so much.
Wonderful talk, as always, Amy. Thank you so much. Let's turn to some of our questions. So a question from Ali Mizra asking, could some of these pathogens be commensals or beneficial in the gut before becoming pathogenic when they enter the bloodstream?
Yeah, that's an absolutely great question. I think a lot of people have brought up this idea of a pathobiont. You know, it's not a bad bug, it's in the wrong place at the wrong time. And I think that that's something that is most certainly the case. There are E. coli that are probably commensal that are making it into the bloodstream, happen to be very flexible and able to tolerate being in the bloodstream, but induce, you know, a sepsis or SARS-like physiology. So absolutely, I do think that there are organisms that are fulfilling that kind of pathobiont category.
Great, thank you. I had a question regarding the Prevotella copri genome that you assembled. Now that you have that assembled genome, presumably your MAG assembly will be an awful lot easier. And have you begun that process? And can you speak to the variants in Prevotella copri in human gut microbiomes? It's a species we're particularly interested in.
Yeah, I mean, so you can use the complete genome that we have to try and do reference-assisted assemblies. I think one of the things that we've started to realize is that the devil is actually in the details, and the locations of these insertion sequences may be incredibly important for driving functions. Quite a few of these insertion sequences have these outwardly directed promoters. And even if they don't have their own outwardly directed promoters, they may be falling into regulatory regions and therefore turning on and off genes. We wonder, for example, if the location of those insertion sequences, it really determines which strains thrive in a certain dietary circumstance. And I can certainly tell you that for a given individual, certainly for the individual from whom we pulled together that genome, the occupancy of the insertion sequences at the different positions is different. So sometimes the insertion sequence is always there, sometimes it's never there, and then we have everything in between. And we've also looked over time, so we know that that also can be variable. So I think it's a way that it kind of adapts.
Mm-hmm. Makes sense. Great, thank you. Well, with that, I will turn over to Peter to round us out. Thanks, Peter.
Okay, thanks so much, everyone. It's been just a remarkable day of talks in science. I think Ami really nicely summarized a lot of the high-level takeaways, but I'll just leave you with sort of three of my own. I think one thing that is hopefully clear from the talks is that the range of approaches and disciplines that are going on now in this area of microbiome research is really broad. So we've heard about both chemistry, molecular biology, and clinical research approaches. There's also this trend now towards much greater precision than what was even thought of 5 or 10 years ago. You know, where originally we might have talked about high-level taxa, we're now talking about strains. We're focusing on single nucleotide polymorphisms instead of genes or gene families. I'm going from compound classes to substrate specificity, or, you know, as Carlito mentioned, you know, taking a macronutrient class like carbohydrate and really drilling down to specific structures and functions. And then finally, I think something that, you know, maybe calls into question the whole organization of the symposium, but I think what's really clear from a number of the talks is that we can't really think about early life in isolation, and that, you know, it's part of this sort of ongoing spectrum that goes into adulthood within a given individual, but also dates back way before we're even born to our parents and our ancestors, and, you know, maybe, as Ruth showed, all the way back to non-human primates. And so I think that really changes at least how I think about the early life microbiome and what sorts of disciplines are going to be needed to understand it. And so with that, I really want to thank the speakers again, our technical support from Brian Coe and ETS, the BCMM committee, and perhaps most importantly, Kristen Dolan and Sarah Mortley, our administrative support for this meeting. I mean, we could not have done any of this without you guys. And so, you know, thank you so much for making this meeting so easy to do in such a great time. And so with that, I'll release you guys, and, you know, I hope you get to go outside and go for a jog or a walk or something after all this Zoom. But stay healthy, and I hope to see you again in person once the pandemic is over.